So at any rate, welcome to our annual symposium. It's really great to see everybody here. I'm Dougie, I'm the director, and it's my privilege to give you the state of the Masonic Cancer Center. I really love to do this talk and present the highlights of the year, but I also face a lot of trepidation in doing this because I know I'm not going to mention some important discoveries that have come from your work. So I apologize in advance if I don't talk about your stuff, but be sure to tell me how I should have talked about your stuff. So I'll apologize in advance. So I know there are people that are new here. I know there are people that are sitting in the audience who have been involved with the Cancer Center since day one, but I wanted to give a little background about the Cancer Center. First off, the Cancer Center mechanism exists because in 1971, President Nixon signed the National Cancer Act, which was commonly referred to as the War on Cancer. And the reason this act was signed was to try to provide additional federal resources to put centers together, including cancer centers, to bring the expertise of those centers to think about the problem of cancer. So our history as a cancer center, we were created by the University and the Board of Regents in 1991. Dr. Lebien, who is here, is probably the only person, maybe, who was involved with that initial effort. He was deputy director since the beginning. He and the late Dr. Kersey received NCI designation status in 1998. So the first time in, we were a comprehensive cancer center. We are now one of 56. We have renewed this every 5 years. Most recently this year, and I'll spend some time talking about that, there are over 600 members of the various schools and colleges throughout the university, which I think is an important strength. For those of you that are new to the Masonic Cancer Center, you may wonder why we're named the Masonic Cancer Center, and that's because the Masons have provided support for cancer research since the mid-50s, but in 2008, they increased their pledge to $65 million, and it was more than appropriate to name the center Masonic Cancer Center. Then we are one of two cancer centers within the state, us and our colleagues down the road. So just a few words about the Masonic Cancer Center. As I mentioned, we had our site visit and grant renewal in May. I want to use a few slides that I showed our site visitors. We are the University of Minnesota. We're a land-grant university and serving the entire state. We have campuses at Duluth and the Hormel Institute that are important research partners. And as you know, we are built within the traditional, ancestral, and contemporary homelands of the Dakota people. While those are words, it's important to recognize this fact, and I hope throughout the course of the day, today at this meeting, as well as some of the remarks I'll make, I'll tell you how we feel this is an important part of our mission. We have to define our catchment area, which is a term of art and really means who do we serve in the state. We aim to serve the entire state of Minnesota, and we are importantly recognizing plans to address underrepresented populations, rural populations, Native Americans, and new immigrants, which are all important constituents of our catchment area. I wanted to say a few words about the foundations of the Cancer Center planning and structure. So I think cancer research is both focused and multidisciplinary. Now that seemed like contradictory terms, but what I really mean by that is we need the expertise that you bring to a problem to think about how to solve it. At the same time, you need to work with your colleagues who may have different levels of expertise to try to solve the same problem. So our program membership is based on scientific disciplines within members from the entire university. At the same time, as a clinician, I'm a breast cancer medical oncologist. I know patients come in through a specific disease door. So we have created the translational working groups, which I'll explain in a minute, to enhance collaboration across the disciplines. So as a breast cancer oncologist, I know I need my colleagues in immunology, in genomics, in population science to help me solve the problem of breast cancer. It just doesn't live within a specific discipline. We also know cancer advances require partnerships outside of the university. I'm not going to say too much today about the Minnesota Cancer Clinical Trials Network. They have a table around the corner. I think you should take a look at it if you want to be involved. And we have a very strong Office of Community Outreach Engagement. And then finally, translational research requires dedicated leadership structure and resources. We have created the Cancer Research and Translational Initiative to try to address that. So this is our program structure. You are all members, or you or your mentors or lab leaders are members of a scientific program. I'm not going to go over each one of them, but as I mentioned, the translational working groups are another structure in which our members interact as a way to solve the problem of cancer. So, how are cancer centers reviewed? We have to have breadth and depth of research organized in scientific programs, as I mentioned. We have to have a collaborative environment with bench to bedside, back to bench, and probably should say population to bedside to population focus. We need to have outreach to our community with bidirectional input, and what I mean by that is we need to understand the problems in our catchment area, and we need to understand how those problems can shape our own scientific endeavors. We need to have a strong research infrastructure, our shared resources, our clinical trials office, and there are 6 essential characteristics, which is, again, this inside baseball part of how core grants are renewed. I'm only going to talk about cancer focus because our members really define what our cancer focus is, which means how much is this cancer center really focused on cancer, which seems like a dumb thing to say, but that's really what they want to know. So how do you measure your cancer focus? One way is to look at research funding. This is the research funding in 2018 at our previous renewal. And it shows you the level of increased funding since June of this year. The green bar shows the National Cancer Institute funding, which went up $5 million. The bluish bar, the dark blue bar, is the NIH peer-reviewed funding, which went up another $4 million. So 20% increased renewal funding since our last renewal, and that's you. That's congratulations to you. Everybody has been working hard. We've had increased funding. As you know, in the era of flat federal funding for peer-reviewed research, a 20% increase is quite an accomplishment, so I applaud you for doing that. The other way to look at advances in publications, these are our publication metrics that were shown at the time of site visit, which is roughly May. The one thing I want to point out, and I don't think I got the pointer thingy, or maybe, no, I don't, but the bottom bar, there's a 24% high impact paper, 25% of the papers we publish are in high impact journals, which is defined as the impact factor greater than 10%, and that's been great too. The other part about Cancer Focus is, do you enroll patients and subjects on interventional trials? This is our 5-year interventional trial accrual comparison. It shows almost 15,000 subjects and patients were enrolled on trials during our last period of support, and that's a 27% increase. So all of these things I show you are what you are doing, and it's really a great honor for me to represent your good work here and also at the Cancer Center site visit. So how did we do during our renewal? We received an overall merit descriptor of outstanding, which is 24, which is our best numerical score ever. I know they use folks that live in the R01 world, you can say 24, well, that's sort of okay, but for Cancer Center grants, that's a pretty good score. Several statements from the critique are, we're making important advances to improve cancer care for the population of the state. We have strong basic translational and clinical research impacting their catchment area needs. Areas for improvement include increased engagement efforts for minority and rural populations, and we'll talk about that a little bit, and cancer screening and clinical trials, and the need for additional efforts to train and equip junior faculty. Overall, the Cancer Center is on a stable trajectory with its science and potential for translational impact, placing the center in a position to reach its full potential. And under the strong leadership of the center director, and with the continued commitment of the institution, the center is well positioned to move to the next level. So that last statement really is not about me. It is about you and the work that you do, and the fact that we organized it in the way that we presented to our site visitors that showed you we were growing and fulfilling our needs. So I want to thank you, congratulate all of you for this successful renewal. So what do we need to think about? Where do we need to go from here? And here's where I'm going to show some examples that I just happened to pick out that I think are kind of interesting and important areas for our center. First, we need to understand causes of cancer and reduce risk. We've had a very strong environmental exposures program around tobacco, now food and others. We need to understand the internal causes of cancer. How do we generate cancers on our own, either through our microbiome or deficient DNA repair pathways? As a clinician, I know we need to improve cancer therapies based on tumor biology. We must eliminate disparities in outcomes for underrepresented groups. We're not making advances as a center unless it benefit all groups that we serve. We should enhance the quality of life for cancer survivors. So I want to pick up just one or two examples of each of these, and you'll hear more about them during the course of the day. The first example, I'm not going to spend a lot of time on because you're going to hear Dr. Pointer talk about the 10,000 family study. The thing I wanted to point out about the 10,000 family study is you can draw a direct line between the Minnesota Masonic Charities gift, the establishment of the 10,000 family registry. They also have a table over there, so take a look, and the ability to successfully compete for this grant. This grant, as Dr. Pointer will tell you, is really designed at looking at some unique exposures within the state and seeing if they explain our increased incidence of leukemia and lymphoma. So they will tell you a little bit more about that. But you could say, well, okay, so fine. We identified exposures. How do we reduce the risk of exposures if you know you're at higher risk? So I wanted to point out the work of Dr. Hecht and Dr. Hatsukami, who are both here. So if I completely misrepresent this study, you feel free to ask them about what they're really doing. But they're looking at a clinical trial of watercress to detoxify environmental carcinogens. The goal is to form a clinical study of watercress to determine if it can enhance the detoxification of common environmental toxicants and carcinogens in both non-smokers and cigarette smokers. So the intervention is this. How many people in the room have drank this? Is it good? Is it something we can sell at Jamba Juice when it works? This is the recipe. This is what it looks like, and the goal is to increase PEITC in the diet through this. I guess this is technically a supplement. This is the trial design, randomized people to the intervention or placebo, 4-week washout, and crossover. And the idea that you can increase the PEITC levels significantly by the watercress intervention is shown here. The washout period shows it does go away after 4 weeks. So the idea that, well, this could work. It could be a way to induce exposures if you have them. And the picture on the cover of Cancer Prevention Research is the watercress itself. And I know, actually, I should have asked you guys how much watercress does it take to make that drink, but it's a lot. No, not so much. Okay. I want to turn to improving cancer therapies based on tumor biology as a next advance. I think most of you know Dr. Miller, and I'm not going to spend a long time on this project because Dr. Zorko has a flash talk a little bit later. But the idea is maybe we can use natural killer cells to target specific epitopes that are more commonly expressed on tumor cells. This happens to be the model of B7H3. You will note in the little diagram, there's a figure that contains a CAM-CD16, so it's a camelid single chain antibody against CD16, which is an activation marker on NK cells. There's an IL-15 molecule which stimulates NK cell proliferation, and then there's another camelid antibody against B7H3. The idea in preclinical studies, this can kill tumor cells by directing NK cells to the tumor cell is an important, interesting advance, and the goal is to bring this forward in a clinical trial. So this is a picture of Dr. Antonarakis and Dr. Zorko at a meeting last week that I asked Dr. Zorko for a slide, and he said, I'm busy, I'm sitting next to Emmanuel trying to get some money out of the Penn State Cancer Foundation, but they sent me the picture. And you'll hear more from Dr. Zorko a little bit later today. We're making this clinical grade. We have FDA pre-investigation meetings already submitted. Hopefully they'll return that and plan for an IND filing in the first quarter with a first-in-human phase 1 and 2 trial here. So when I said to you, one important thing that cancer centers must do is translate from bench to bedside. This is the example I am speaking about, and we do have a sponsor, GT Biopharma, to hear this, and you'll hear more to support this, and you'll hear more from Dr. Zorko in a minute. So I decided to be self-indulgent and talk about something I've been interested. I think ever since I've been giving this talk, I've actually never talked about what I've been interested in, but I'll talk about breast cancer therapy with the idea that if we give breast cancer therapy before surgery, called neoadjuvant therapy, can observe the complete disappearance of the tumor. And we were able to show within this clinical trial that complete disappearance of the tumor, pathological complete responses associated with better long-term outcomes. And beyond that, therapy could be adjusted if you're not heading toward a pathological complete response. And I think biomarkers could move us to precision therapy. This is a trial that we've been involved with since its day one. It's called the I-SPY2 trial. For those of you who remember our keynote Kennedy lecture last year, Dr. Esserman, she talked about I-SPY2 as part of what she's doing. But the idea is we can give therapy before treatment, and then, let me see if I can get my marker. Okay, there it is. We can give therapy before patients go to surgery and identify pathologic complete response, and we can give experimental agents in combination with standard of care therapy. The reason I wanted to bring this up is there are over 30 centers. We are the 4th leading accrual center in the country. This is obviously another example of team science, or where the team really has to work together. We have our patients, a radiologist, everybody on that list is responsible for preparing our accrual site, our leading accrual. If you notice, one of my career goals is to accrue at a higher level in the Mayo Clinic in Rochester, and so they're right behind us, so it is neck and neck, but so far we're achieving that goal. Here's my disclaimer. So I've been chair of what's called the Agents Selection Committee for the past 10 plus years. These are all the drugs in the trial. I know it's fine print, but almost all our sponsors have contributed the drug to this trial. They've either been better than the standard of care, which means it's graduated, they've been the same, or they've been stopped for toxicity, or they're ongoing. So this is a standing platform phase 2 trial. I actually had the opportunity to work on a drug, ganitumab, which is an anti-IGF receptor inhibitor, which is something we work on, which was not better than control, but I have about another hour talk on how this trial actually showed something important about biomarkers related to its effectiveness, but I'm not going to talk to you about that today. What I am going to talk to you is about these 2 drugs. This one happens to be veliparib, a PARP inhibitor, and carboplatin, as well as pembrolizumab, the immune checkpoint inhibitor. About a year ago, we published the idea that you could look at biomarkers, meaning RNA expression profiling and reverse phase protein arrays of patients prior to treatment to predict better outcomes, and this was published in Cancer Cell. So what this shows is that for patients who have the triple negative subtype, in other words, not HER2 or hormone receptor positive, about 2/3 of them have an immune positive signature as defined by expression profiling, and over almost 90% of those patients have complete pathologic response to pembro. In contrast, there are some triple negatives that have DNA damage response signatures, again, about 2/3 of those. They have a benefit from the addition of veliparib and carboplatin. There are some that have none. If you have none of the above, you don't benefit from these drugs. In contrast, if you have both, you have a very high response rate. So that tells you if we're going to give therapies, and in particular we're going to give immune therapies, we may have a way to distinguish between these patients who really need it because they have an immune positive signature, as opposed to these patients who suffer toxicities, are no better than control, but need a different kind of chemotherapy. And for those of you that work in this disease, patients who have response to neither of these, and we have not solved that yet. So that's one way that our participation and the team participation is hopefully moving the needle on precision medicine. I want to say a word about disparities and outcomes for underrepresented groups. We just recently applied for a supplement to strengthen research, training, and outreach capacity. Drs. Gebra and Pannell put in a supplement to recruit American Indian Training Coordinator. This is a collaboration with us and the Clinical Trials Network, as well as the Center for American Indian Minority Health in Duluth. And the American Indian Training Navigator will develop a strategy to identify barriers and opportunities to consider and take action towards cancer career for Native communities. So the idea that we need to have a more inclusive and group of investigators as well as patients enrolled on trial is an important part of what we should be doing as a cancer center. I do want to comment, these were the centers that were funded. There are roughly 70 applications. I think almost every cancer center applied for this, and I want to give credit to Dr. Gabriel Pinel for receiving this. I want to end on the last part, enhance quality of life for cancer survivors, and just feature a project that Drs. Vogel and Gupta are doing to address something that I haven't thought about until I had a good discussion with Dr. Gupta and Vogel about time toxicity. So time toxicity relates to how much of a patient's life is spent doing things related to their therapy. So I'm not going to go over these studies in detail except to tell you it is a novel measure for capturing the burden of cancer treatment. Dr. Vogel has a cell phone-based app to go ahead and try to look at this and capture the data, where Dr. Gupta gives you an example of the time toxicity we require of patients who spend significant amount of their life getting care that we deliver. So with those examples, I want to talk about how do we defeat cancer. We need to support research to make paradigm, policy, and practice-changing discoveries. We need to develop research teams to solve hard problems and translate discoveries to reduce cancer burden. We have to engage our partners in our mission, which include people at risk for cancer, people with a cancer diagnosis, cancer advocates and supporters, and most importantly, you, the researchers who make this possible. Again, I'm really thrilled to be part of representing the hard work that you have done, and I think it's hopefully reassuring and gratifying to you as it is to me that a group of our peers have also endorsed the work that you're doing. I want to finally thank the symposium planners who put this together and has really done a stellar job of the program throughout the course of the day. And again, I want to thank you for all the hard work that you've done. I have said, and many of the people on the leadership team know this, Dr. Kersey did 2 renewals. I have done 4 renewals. I think that shows that Dr. Kersey was smarter than I was and got out earlier than I did. But I think it's time, as Dr. Kersey told me, to have a new and improved director. So if everything goes as forward, I'm planning to step down from this position. Hopefully next year at this time, there'll be a new cancer center director. I'm not going anywhere. My lab is just looking a little worried at the moment. I'm gonna keep working in the lab. I'm going to keep working in the clinic, but I'm looking forward to helping transition this fabulous and fantastic, and those are not adjectival descriptors by the NCI. I think they should be fabulous and fantastic cancer center to our next generation. So again, I really want to thank you. It's really a privilege and an honor to be the cancer center director for now 16 plus years, and I really appreciate the opportunity to lead this stellar group. So with that, I do want to introduce the B.J. Kennedy Lectureship in Oncology. For those of you who were fortunate to have known B.J. Kennedy, you knew a little bit about his importance to this university. He received his MD from the University of Minnesota. He did his postgraduate training around the country and in Canada. And then in 1952, he returned to the University of Minnesota. Dr. Kennedy was one of the first to recognize the need for specialized management of cancer care. He also advocated for specialized care for the construction of the Masonic Memorial Hospital. Dr. Kennedy was the first to make contact with the Masons. Some of you, most of you, many of you have been here long enough to know where the Masonic Memorial Hospital was. It's where the new education building is, but certainly the idea that a cancer hospital was necessary was really a pioneering thought behind Dr. Kennedy, and he pioneered the medical therapy for cancer. So this is one of our 2 named lectureships, and I will let Dr. Hatsukami introduce this year's B.J. Kennedy lecture. So again, thank you, and turn it over to Dr. Hatsukami.
Well, good morning, everyone. I'm really, truly delighted that Dr. Electra Pascat accepted our invitation to give the B.J. Kennedy lecture this year. Just as Dr. Kennedy was a pioneer in forging the discipline of medical oncology, I think Electra Pascat has been a pioneer and a leader in shining a light on cancer health disparities among populations that are underserved. Dr. Pascat holds a dizzying a number of leadership roles. She is professor and the director of the Division of Cancer Prevention and Control in the College of Medicine at The Ohio State University. And she also holds the Marion and Rowley Designated Chair for Cancer Research. She is also a professor in the College of Public Health. For the Ohio State University Comprehensive Cancer Center, the James, she is the Associate Director for Population Sciences and Community Outreach and is the founding and current director of the Center for Cancer Health Equity. Additionally, she holds a number of leadership roles in organizations outside her institution, and I will only name a few. She was past president of ASPO, the American Society of Preventative Oncology, and she oversees the ASPO Associate Director and Program Leader Workshop. Because of her tremendous contributions to this organization, a scholarship has been named in her honor. She was recently a member of the National Cancer Advisory Board, where she co-chaired an ad hoc subcommittee on population science, epidemiology, and disparities. She is a current member of the National Academy's Ad Hoc Committee on National Strategies for Cancer Control. She is the director of the Cancer Control Program in the organization Alliance, and she is also a member of the Ohio's Commission on Minority Health. Dr. Pascat is widely known and highly respected for her research in cancer health disparities. Her research focus has been mainly on cancer prevention intervention, cancer detection, access to diagnostics and treatment, and survivorship issues, especially or specifically among underserved populations. She is multiple PI on a recently funded Moonshot initiative that is looking at improving colorectal cancer screening through implementation science. She also is a multiple PI on a PO that is looking at improving cervical cancer prevention services in Appalachia. And finally, she is also a multiple PI on the Women's Health Initiative Cancer Survivor Study. So you can see that she is a role model to all of us. And so without further delay, I present to you Dr. Electra Pascat.
Thank you so much for that wonderful introduction, Dorothy, and Dr. Yee, thank you for that presentation. It is not an easy job to be a cancer center director, and I have assisted in putting together 7 CCSG applications, and they are not for the faint-hearted, but I think you're just doing a fabulous job, and I think fabulous is a perfectly good adjective to use. While we're getting the slides up, I'll just tell you a little bit of what I'm going to talk about today. Dr. Yee talked about new paradigms, changing paradigms. So my presentation, I'm going to show you sort of a patchwork that I'd like you to put together in your own quilt to think about how we could work together across basic, clinical, and population sciences to actually reduce disparities. And I think Dr. Yi, in his presentation, really wanted to get that point across, that we do need to do translational research in order to make an impact. Thank you. So, okay, glasses are good. Oops. Okay, so we're going to talk about the role of place, stress, and aging. And again, keep an open mind and see if you can put all the pieces together. These are my disclosures. I don't believe any of these are going to be applicable to my presentation today. So this is sort of the undergirding or the key thesis of my presentation. I am an epidemiologist, and in epidemiology, we study epidemics and how determinants, meaning the different distribution, different descriptives of a disease, are distributed in a population. We all know age is a risk factor for cancer. That's universal across genders, age. And it's because the aging of cells increases the risk for cancer to develop. So are there populations that physiologically age prematurely? And are disparities then in cancer outcomes caused by this phenomenon of premature physiological aging? So that's the undergirding sort of questioning that led me to put this talk together. Okay, was I behind one? Sorry. So the key messages will be, first of all, that there are disparities in cancer outcomes, and I want us to look at the impact of place, okay, where you live, for example. Stress can be mental, social, or environmental, and we're going to go through some examples of these. And the impact of this stress gets under the skin and causes this premature aging of cells, impairs the immune function, for example, deters repair mechanisms, and it causes cancer, and in many cases, bad cancer. I'll give you some examples of that, which lead then to disparities. So let's start a little bit with some pictures. This is what we do in epidemiology. So this is a picture of death rates, mortality rates for all cancers, all races. And the hot spots are in red. So you see the hot spots there in the Appalachian and the South area. So let's now turn to an example of lung cancer. And the figure on your left are lung cancer death rates by county. Where are the highest ones? Do you see some parallel to the previous map I showed you? Heavily concentrated in that Appalachian area in the South. And then if you look on your right, this is the prevalence of current cigarette smoking and smokeless tobacco among the US. Are you seeing sort of patterns mapping on the same place? Okay, then if we go and look at the distribution of tobacco farms, similar places have the highest areas, the South and the Appalachian areas. And then this last figure here on the right is the state spending on tobacco prevention. And red are the states with the lowest percentage. Are you seeing the same states sort of map in the high area? So that leads me to talk about my favorite model to understand health disparities. This is a model we developed in the Centers for Population Health and Health Disparities. The lead author was Dick Warneke, and we lovingly call this the Warneke model. Dick passed away suddenly last year. But this is one of the socioecologic models, which basically says the reason that we have disparities is not just because a patient smokes, for example. There are interacting factors that go from biology all the way up to social conditions and policies that all work together to cause disparities. So if we think about the lung cancer model, we know that Blacks or African Americans are more prone to like menthol cigarettes. Okay, is it a biological phenomenon interacting with individual risk factors? And then those of you who are smokologists, as my friend Betty Thompson calls herself, you know that if people are around other people who smoke, it's harder to get them to quit. That's the social relationships. And if we think about the social and physical context, then we know that the tobacco companies market more heavily in areas that are poor and underserved. In Appalachia, we've actually done surveys of the size and the number of the the tobacco ads there. And the same in areas where African American Blacks live. The advertising is specifically targeted to them. Institutions, do they frequent institutions that don't talk about smoking cessation? I can tell you that's true in Appalachia. That's one of our projects right now, is try to teach clinicians how to talk about smoking cessation. And then the social conditions and policies, those were the last two graphs I showed you, with the state monies going to cessation services and then being in a farming state. So they all interact with each other to then cause disparities. I love this model because it's easy to apply, not only for trying to understand why you see disparities, but then if you know the why, then you can design interventions to change that picture. And we're doing this in many examples. But I will use this model later on. So let's move for now a minute. We're still trying to build the issues associated with my thesis. And let's talk about the theoretical basis for social stress and cancer. And this is a model published by Dina Boberg. And does this, explain place associations? Well, if, the person is the little head there, and there are stressors that people face from the overarching contextual issues. For example, in society, discrimination, stressful work environments, in the family, marital status, or a caregiving role, and at the individual level, as shown there by demographics, for example, or BMI. Those stressors, then, we know that the central nervous system perceives them, and then we have affect, cognition, decision-making, behavioral actions, et cetera. And we have this perceived stress that can cause depression and anxiety. And all of that actually it affects the autonomic and neuroendocrine pathways and interacts with behaviors, either risk-promoting behaviors. For example, when people feel stressed, they smoke, they drink. In our Appalachian population, we see increased risky sexual behaviors as coping mechanisms for the everyday stressors they face in their life. And then those, both the biology and the behavior, can impact cancer development. And you can see there the cancer control continuum from cancer development, detection, diagnosis, treatment, all the way to survivorship care and supportive care. So this model sort of explains, actually physiologically, the parts of the model that I just showed you before. So then the effects of stress-associated factors on the tumor microenvironment are discussed here. So responses to stressors involve CNS perceptions of threat and subsequent activity of the autonomic nervous system and the hypothalamic pituitary adrenal axis. stress hormones are released, and they modulate the activity of multiple components in the tumor microenvironment. And these effects include promotion of tumor cell growth, migration, invasive capacity angiogenesis. They can activate oncogenic viruses, for example, HPV, that might have been dormant for a while. And they can also alter the immune function so that the immune system cannot combat any viruses or abnormal cells. So then the downstream effects create a permissive environment for tumor initiation, growth, and progression. And you can see the different factors that are related, including IL-6, MMP, and VEGF. So now I've sort of given you the theory and the biologic rationale that sort of backs this up very strongly. And now I'm going to go into some real-world examples that have been done by our team as well as some of my colleagues across the country. So the first example is one of my favorite examples, and it is one of our sister sites in the Centers for Population Health and Health Disparities, led at the University of Chicago led by Dr. Sarah Gellert and Fumio Olopade. And their research question was, how do the social environments of women explain black-white disparities in breast cancer mortality? And this is in Chicago, and you can see the disparity. Disparity is a difference that should not exist, okay? So this is a difference in death rates. For white women, you can see in the gray, and then black women in black have a significantly higher death rate from breast cancer. If there was no disparity, we would be at the first part of the lines, and that is really where we should be. So their center had 4 projects, and it goes along that Wernicke model I showed you from biology all the way up to community in the neighborhoods. And so they did have animal model studies as well as out in the communities in the south side of Chicago with the black women there, and they surveyed them and collected biological measurements as well as looked at their environmental exposures in the community, looked at crime, for example, housing, physical environment. So this was a real cool experiment. To somebody who's a population scientist, I just think this is really cool. So they had litter mates, and they separated them into grouped and isolated. And then they looked at the tumor burden and found a significantly higher breast cancer tumor burden in the top right panel in the isolated female animals versus the group housed. And then they looked at the endocrine stress response. And for the animals, the stress was both that they could move them or they did a restraint. And if you'll notice, the bottom graph is the restraint as the stressor. They were pretty similar at baseline, but then after the restraint, the group housed in the red lines, the stress response, endocrine stress response went up and it came back down. But if you can see the blue group in the isolated ones, they did not come back down, which means that they concluded that the hormone response is a conduit from social environment or stressors to gene expression. So that this little figure here, the social isolation caused acquired vigilance with a hormone response, which contributed to more aggressive tumors. And that poor little animal there on the bottom left doesn't look so great. But really, they actually had, I'll show you these cages they had, that they could actually see what their patterns were. So the isolated animals there on the far left, they developed a vigilant pattern of patrolling their cage. So it was patrolling the perimeter, right? Whereas the group house, they just went all over, you know, they groomed each other and everything. And so they paralleled that to what they saw on the south side of Chicago. And they saw race associated with poverty, disruption, neighborhood crime, and fractured community. And the women there they felt isolated. And they had acquired vigilance and depression, which affected their stress hormone dynamics and cell survival and tumor development. And the really bad cancers they developed were the triple negative breast cancers. And this is an example here in Black women on the south side of Chicago, how the fear, the crime, the isolation really caused this. And now I'll tell you a little bit about our project. We were in the Centers for Population Health and Health Disparities, and the project we tackled was cervical cancer. Now, we titled this a little aside for you, so you can tell just a little bit about Appalachia. We wrote this grant, Reducing Cervical Cancer in Appalachia, and then we went out and started doing our pilot work, talking to people, to women, to communities, and we found, oh, you cannot say cancer in Appalachia, because that is a death sentence. They fear cancer. And then you can't say cervix, because our kids don't have sex. You know, that's not a word. So how do you do a study? And we were funded for 2 cycles. How do you do a study without mentioning cervical cancer? On cervical cancer, you don't mention it. So we called it CARE, Community Awareness, Resources, and Education. And a little bit about Appalachia, for those of you not familiar. The red area there is, this map is the mortality, death rates by state for cervical cancer in white women. And this was the map when I moved to Ohio State in 2002. And the red area maps pretty well on the geographic definition of Appalachia, along the Appalachian Mountains in the United States. And what we soon found in terms of why we saw elevated cancer rates in Appalachian Ohio is that very high abnormal pap test rates, high smoking prevalence, high just among women, but among women with abnormal paps, the smoking prevalence was 68%. High rates of HPV. We were one of the first to actually quantify the prevalence of HPV in Appalachian populations. It is higher than that in the US. High rates of poverty, and as I mentioned earlier, high rates of risky sexual behavior, early age of first intercourse, and multiple sexual partners, which they use to deal with the stress. And yes, our Appalachian women also did say they felt isolated, afraid to go out of their homes because of the crime. Very similar to what we were hearing in the south side of Chicago. And the result of that is high rates of depression. I was astounded when we did the CESD in these young women. CSD is a screener that indicates a person needs to follow up for a more thorough diagnosis and treatment of depression if they scored a certain level. And in this young population, the rates were actually greater than what we have found when we have been doing studies of cancer survivors. So very high depression rates. And at the same time, really low healthcare access. Just two facts for you. In our 32 Appalachian County area, we have 5 or 7 counties that have no mammography facilities. So we bring our mammogram van there. And then we have about 5 counties that have no local hospital. And you might say, well, maybe that's good because of the care. But anyway, they don't have healthcare access. And surprisingly, we found low healthcare provider trust. And all of this is framed within the Appalachian culture. So our first grant, first center, we examined smoking, we looked at increasing screening, and we did our assessment of the prevalence of HPV. So we were very successful in increasing the uptake of Pap tests. We were successful in smoking cessation in 3 and 6 months. By 12 months, the difference had disappeared, significant difference had disappeared. And as I mentioned, we qualified the prevalence of HPV in Appalachia. So based on our research for the first 5 years, we developed this model of stress and HPV infection, which says that because of where the women lived, and there was low socioeconomic status, low healthcare access, loneliness, or very little social support. And we thought they might be sort of buffered by having an Appalachian self-identity, similar to what others have written about the Hispanic population. We didn't see that. All the women we surveyed lived in Appalachian area, and only about 1/3 of them said that they were Appalachian. They see themselves as either residents of their county or of their state. They are very proud to be an Ohioan. And then that causes perceived stress. In addition, they have poor coping strategies. And the way that they deal with this stress is smoking, alcohol, and the risky health behaviors. But both the smoking, alcohol, and the stress cause depressive symptoms, which then impact the immune system. And we were very concerned that with the risky sexual behaviors, repeated HPV exposure, and an infection, would the immune system be responsive to the HPV vaccine in order to stop the progression, the persistence, and the progression to cervical cancer? So when we put in our renewal for the center, this was our model, linking the 4 projects. And it's still the Warnke model, it's just rotated 45 degrees. And our first project was a gene-environment interaction project, and the second looked at social networks and the effects of social networks on women's smoking patterns. Project 3 was a psychoneuroimmunology study where we actually gave the vaccine and saw if it had if it was still start all over again. We gave the vaccine and we looked at perceived stress levels and saw if there was a good response to the vaccine, irrespective of the stress levels. And then Project 4 was our randomized controlled trial to increase the uptake of HPV vaccine in young girls. It was only approved for girls at the time we did this. So after our 5 to 7 years of doing this project, we developed our own socioecologic model for why we see cervical cancer disparities in Appalachia. And at the bottom, the genetic mechanisms, we actually found some association we see between some genetic mechanisms, the development of cervical cancer and smoking status, immune functions, and of course HPV infection, and then individual risk factors, tobacco, age, young age, socioeconomic status, low socioeconomic status, low education, high depression, and then of course lack of screening. Social relationships, social networks, social support, social influence, really big. I'll give you one quick example. When we were doing some focus groups with parents, I don't want to give my daughter, that HPV vaccine, because before I get home from the clinic, my mother will be at my house, and when I enter the door, she will say, "Why did you give your daughter that green ticket to have sex?" So the social networks are very influential. And then we looked at social cohesion, neighborhood disadvantage, of course, the healthcare system, and then poverty and discrimination. And you say, well, if I recall, Appalachia is like 98% white. Yes. But there's other types of discrimination besides racial discrimination. We were doing focus groups for another study with my colleagues in Pennsylvania. And if you remember, the map I showed you of Appalachia region includes Pennsylvania. So we wanted to call this program we were developing, exercise and diet intervention, Living Well Appalachia, something like that. And we got to the Appalachian folks who lived in Pennsylvania. They said, we're not Appalachian. Those people, this is what they said, those people live in Kentucky and West Virginia. They are poor, they work in the coal mines, and they don't have any teeth. Okay, so these were Appalachian, Pennsylvania's people, and they were calling fellow Appalachians, they were describing them, right? Not so nice. And so there is discrimination in our poor and our underserved populations that do impact stress. So now I'm going to go through some other examples of disparities that cause stress, premature aging, and cancer. We're going to talk about extreme population level events, redlining, which is an example of structural racism, intersectionality between race and age, and some neighborhood socioeconomic factors, and then biology, just to show you the multi-level influences on cancer outcomes. So this is a conceptual model of the association between neighborhood and cancer outcomes, which was published by one of my wonderful junior faculty. And it's very similar to the first model that I showed you with the chronic stress in the middle, but this is being measured by allostatic load, for example, and links to molecular cancer biology. But you can see that residential stressors, which I've been skirting around, can be listed in social and built environments. So built environment, for example, physical disorder. In his research, he gets students to go on Google Earth and actually look at neighborhoods and measures physical disorder. Social in terms of socioeconomic deprivation, segregation, and discrimination. And then the health behaviors we've talked about, and then social disadvantage, low socioeconomic status, interpersonal discrimination, et cetera. So the top blue box is more at the higher level, the area level, right? So we do have socioeconomic deprivation at the area level, as well as in the blue box the green box at the individual level. So this is one example of population events, level events, that focused on it's a constant event, discrimination, violence, SES, or extreme population level events could be episodic. For example, the Holocaust, famine, and COVID studies. And several researchers have found higher risk of developing cancer in Holocaust survivors, as you can see there, for all sites in colorectal and lung, and any cancer. And then Elias et al. Did a really interesting study looking at the effect of the Dutch hunger winter and found elevated risk for breast cancer. Byers et al., which I'm going to show you the actual data on, looked at redlining, and this example of discrimination. So this was a very nice study that Kristen Byer published in the Journal of Clinical Oncology to show that redlining, which was a practice that happened a very long time ago, where certain areas were redlined by banks, and those people who lived in those areas were not allowed to get mortgages, or the rates to get mortgages were astronomically high. And the areas tended to be where Black African American residents lived. So you can see here that historic redlining has an impact on both overall survival and also breast cancer specific survival. And this is another study that looked at neighborhood deprivation index. So remember the box on Jesse's model, that was the neighborhood levels. This is neighborhood deprivation. And they looked at this racial disparity in survival outcomes for early hormone receptive positive breast cancer. They adjusted for insurance status and neighborhood deprivation. This was a post-hoc analysis from the Taylor X randomized controlled trial. And what they found was that insurance and neighborhood deprivation index correlated with overall survival, in that Black women had poorer survival. So the top left is the lowest deprivation, and then the bottom right is the highest deprivation. So again, where you live. This is some of the work that the hematologic malignancy group at our institution have been doing, and I want to put a plug in here for the basic and clinical folks to think about working with the pop-sci people. Some of the clinicians started seeing in their clinical practice poorer outcomes in their Black patients. And so they came to us in COE, Community Outreach Engagement, and said, can you help us look at this? So I have a wonderful epidemiologist that works for me, and he is very good with SEER registry data. So they started working with him, and we as a group looked at this and actually found that there were lower survival rates in young Black AML patients. And then they took that fact and they ran with it and went to CLGB Alliance, which is a cooperative group organization, and found the same thing in their studies, and then actually looked at some of the cytogenetics to see if the cytogenetics could explain the differences that they were seeing in survival. And then the same team is moving on to look at zip code-based socioeconomic composition, again in CLGB Alliance, in AML. And the darker colors on the maps are the lowest deprivation, and the lighter green are the highest. So they divided into two deprivation levels, low and high, and they tested for associations with clinical and molecular features. And what they found was non-Hispanic whites, here we go, this is the better, that high socioeconomic deprivation was lower in whites, lower in allogeneic transplants, and lower in these different mutations. And then what they did was they found that higher deprivation, social deprivation, independently predicted survival. So the high deprivation is there in the red bars. And here's the multivariate model for overall survival. Again, the social deprivation index. And then intermediate versus favorable ELN. So in residence areas with high socioeconomic deprivation, it was associated with poor survival. And it predicted outcomes independently from the routinely used molecular-based risk stratification in AML. This is kind of hot stuff here. They just published this this year. But they also found that Black patients still had inferior survival even when restricting analyses to the same socioeconomic status. So even in the lower socioeconomic status, there were disparities by race. So we're continuing to work with this team, but I just want to say they're just lovely to work with, and we're having a great time. So I want to talk a little bit about now, since we've been talking about area deprivation index, how it applies to cancer screening behavior. So we just finished this very large R01, and we worked in rural Ohio and Indiana. And these areas tend to be mainly white, lower income, less educated, have higher breast, cervical, and colorectal cancer incidence and mortality. So area deprivation allows for rankings of neighborhoods by socioeconomic disadvantage at the census block group level. And in creating this measure, it considers income, education, employment, and housing quality using the American Community Survey 5-year estimates. And these are calculated by the University of Wisconsin. So you see the map of the U.S. where Indiana and Ohio are, but next to the middle line on the right, those pink counties are all the rural counties, 98 rural counties, and the cross-hatched area is Appalachia. So we did a study in these 98 rural counties. The bottom shows you where the most disadvantaged block groups are. Those are in the red and the orange, and they pretty much, again, match on to the rural counties, and especially in the Appalachian areas. So our study was called RISE, and what we wanted to do was develop and then test an intervention to improve the uptake of cancer screening for women 50 to 74 who lived in those rural areas. And this is the first intervention ever done that simultaneously tried to increase breast, cervical, and colorectal cancer screening in any population. And what we tested was 2 interventions, a tailored DVD versus the tailored DVD plus telephonic patient navigation. So all interventions were remote. And we also had a usual care arm. The study section made us do usual care. And then we did this all by medical records, also medical record verification, they were eligible, medical verification if they got any screenings. And then we also did some cost-effective analysis. So we recruited from those 98 rural counties I showed you. Here's the eligibility criteria. Why did you do a DVD Electra? That is so backwards. Well, I can tell you, even today, that there are many counties in those pink areas do not have even cell phone access or internet. And we're dealing right now, today, with that issue because there is a dear little clinic, I love them to death, in Quaker City, Ohio. You lose cell service as soon as you get off the highway, and we want to bring the mammogram van there. And we have no internet service. Okay, this is 2023, and I have argued at NCAV, I don't know, you probably weren't there, Dorothy, when I argued that we still have a digital divide today. Okay, off the soapbox. So these are participant guidelines, they did represent the area just a little higher socioeconomic status, probably because we had to recruit through Facebook. Whole nother lecture on that. All right, so what we found was that we did 2 measures, all screenings on the left, which is what proportion of women got up to date with all 3. So they all needed at least 1. Some needed 2, some needed 3 tests. And then any needed. So if you needed a breast and a cervical, did you get either one of them, is the graph here on the right. And you can see that in the highest area deprivation index counties, which is the left bar in the comparison, the rate of screening was significantly lower compared to the higher, the less deprived counties. The odds ratio was 0.58, so significantly lower. And the same with the any needed screenings. And what I have highlighted there in the yellow is that there was no intervention, no interaction with intervention, which means the intervention didn't work any differently in either group. But it tells us that this group needs something else, something else. And I should tell you that we had a very highly statistically significant effect of the combined intervention, almost 6-fold increase in becoming up to date with all 3. This is another study that was published in the Journal of the National Cancer Institute, and it looked at the impact of Medicaid expansion status. And we know that this is looking at changes in overall survival. And there was not only the effect of race, socioeconomic status, and rural, all of those doing worse survival. But if you stratified by if it was a Medicaid expansion state or not, that had an independent effect in that. So this is some cool stuff that was done in another group. So let's go back to the aging idea. We know that tumor biology changes with age in terms of stromal and immunosenescence, inflammatory system aging. Those soldiers, I like to explain the immune system as soldiers when I talk to community groups. And it causes tumor initiation and growth. And so can area level stress replicate these effects of aging? So this was a very nice study where they looked at, in triple negative breast cancer, ancestry-associated gene expression profiles. And they found that these showed population-level distinct immunological landscapes, which could provide a link between African ancestry and the triple negative breast cancer immunological landscape. And then the last examples I'm going to show you come from my friend Christine Ambersoni's group, where they looked at tumor infiltrates in breast tumors from Black and white women, and they found that, yeah, there were higher tumor infiltrating lymphocytes, but there were differences in them, and the ratios aggregate to the absolute CD8 T cell fractions were actually lower in Black women. And so they are now continuing the New York breast cancer study to investigate the immune contexture in tumors from Black and white breast cancer. So there might be higher immune functioning, but it's really not the kind that we need to actually attack the cancer cells. So the last thing I want to leave you with is just a thought. And that thought is, what impact will the COVID-19 pandemic have on cancer incidence and mortality in the future? And this is a figure that shows, again, COVID-19 pandemic could affect breast cancer in terms of those exact factors there. The built environment, for example, we didn't go out and do much physical activity. Natural environment, for example, air pollution, social environment, food availability. I know many of areas, both inner city and out in rural areas, we have food deserts. I can tell you one county in Ohio that didn't have a grocery store for 4 years. And of course, economic, geopolitical environment, healthcare environment, et cetera, and what effect will that have on cancer incidence and mortality? So lastly, my take-home message is this. Social stressors contribute to cancer incidence and mortality. Where you live contributes to social stress. Neighborhood SES, historical patterns of oppression, access to a lot of things, healthy food, for example, and healthcare, discrimination and violence. These are the stressors, and they lead to cancer, which contributes to aging and disparities. So, to improve outcomes for everyone, I think we should be asking, like, as a vital sign, where do you live? And then we can localize actionable risk factors and intervene with access to services, for example, patient navigation, which is really an important intervention that we use at our place. So thank you very much for your attention. I'd just like you to think and ask yourself, where do you live and where do your patients live? Thanks very much.
Awesome. Well, thank you very much, Dr. Pasquette, for the fantastic presentation. And thank you, everybody, for being here. We appreciate you. So we're going to open up the floor for some Q&A. We'll have 2 microphones being passed around for anybody who has questions, so just make sure to flag them down. And then when you're speaking, to try to speak nice and clear into the microphone, close to your mouth, so that we can hear you and so our online audience can also hear you. Cool. And then we will also be taking some questions from the people on Zoom. So for everyone out there watching, make sure you type up some questions and we'll take them at the end. All right. I think I need to do This one's yours.
You have excellent presentation. So I have a question. Let's take the AML studies that you showed difference between African Americans and others with respect to outcome. You know, when you think about African American populations and their relationship to healthcare systems, there's a couple of variables. One is their actual access and the type of insurance or lack thereof that they have. And the other variable is trust and mistrust. And can you separate those two when you look at those differences in the curve?
So those studies were not done with any interviews with the patients. So we don't have any individual information. And the questions you ask are very appropriate to inquiring about trust, inquiring about access. We could look at proxy measures in terms of time to diagnosis. We were able to go back to the original hospital where they were seen to see time from onset of symptoms to diagnosis and things like that. But you are spot on. Not all of our populations have access to a comprehensive cancer center. And you know, I see the access issue myself as a patient. You know, I'm a 3-time breast cancer survivor, and I often, when I had gotten my treatments, I was like, wow, you know, I'm a faculty member. I know a little bit about this. Can you imagine poor Irma Jones off the street not realizing that when you get radiation treatments, okay, this back in the day, guys, that you have to come every day for 6 weeks, the same time, and they mark you with marker that you can't wash, that they tell you can't use soap, wear a bra, or use deodorant. You know, how is the woman, you know, the actual woman on the street going to understand that? And then you have insurance problems, which don't get me going. You know, I'm very well insured, and I still have denials. So who has the time to deal with all that? We use navigators a lot and community health workers, and they're from the communities that we serve in the state of Ohio, which is our catchment area. So that our community health workers are trusted because they come from those communities. And when they bring, you know, a Somali woman who ran out of her chemotherapy appointment with the infusion needle in, but ran out because nobody had told her what was going out. This was at another hospital. And, you know, my wonderful community health worker found her, calmed her down, talked her down, and got her into, you know, the James to finish her chemotherapy. And the woman is still alive today. But that's what we need. We need the human touch. And I think we've forgotten that.
I have a question back here. Really interesting, thanks. I'm kind of focusing on, I think you hit some things on depression, isolation. Are you starting to capture any data on social media use as it maybe relates to some of those things?
Yeah, that's a really good question. And we did some of that in our focus groups in terms of HPV vaccination. And we did collect some of that data in our more recent studies looking at COVID and vaccination behaviors, testing behaviors. So we are analyzing that, as well as even access to telehealth. You know, when you talk about access, and a lot of our populations don't have that. So we are looking at that. But we know from other studies, social media is a huge influencer. Yeah, thanks. Dorothy.
Lecter, there was a real fabulous presentation really showing us how social deprivation can influence cancer outcomes. I guess my question is, what do you envision in 10 years from now in terms of reducing cancer health disparities?
Right, so my vision would be that when we are seeing patients, whether it's primary care or in oncology clinics, that we ask the social determinants of health, we ask where they live, and then based on the responses, we are able to intervene with appropriate help through navigators. Sorry, I do like navigators. I think they are wonderful, and knock wood, the CMS rule will go into effect in January, the end of January, and we'll be able actually to bill and get reimbursed for navigation services. But I think everybody deserves a navigator. And the reason I like navigators is because my barriers are different from your barriers. My needs are different from your needs. My culture, my history is different from your history. And the navigator can be personalized enough to deal with that. So I think that is one big step. And if I could also, since I have my soapbox, I could say, you know, there is one cancer that we could start eliminating. Not just eliminate disparities, but eliminate. Does anybody know what that cancer is? If we all, if as a collective group in the United States got together, we could eliminate this cancer maybe in 6 years or so, 6 to 8 years. Anybody guess what that cancer is? I hear it, say it louder. Cervical, yes. And the WHO has the 3 pillars, and they put that challenge out to eliminate cervical cancer. Eliminate is not the same as eradicate. Okay, eliminate is get it to the low, very low population level, so that it's not, you know, causing such problems. 4 per 100,000 is what the smart people say. 3 pillars: HPV vaccination, cervical screening, and follow-up on abnormalities. That last one, that's what I did my doctoral dissertation on. Graduated in '89. The rate of follow-up, appropriate and timely follow-up at that time was 60%. Do you know what it is now? 60%. Travesty. This is low-hanging fruit, folks. And if we can prove as a scientific community that we can eliminate one cancer, you know, all our cancer centers, mine included, say we're going to end cancer. You know, how are we going to do that? We could start with cervical cancer, but nobody wants to touch it because stigma. You know, I had an cancer epidemiology professor, and I went to a very nice school. When he got to cervical cancer, he said, only prostitutes get cervical cancer now. Unfortunately, that's a mindset still today. It's not right. My mother had cervical cancer. That was her first of 4 cancers. Okay. So we can do it, but there's a lot of factors. So.
Yep. Thanks, Electra and Blaze. I really appreciated your presentation. So I have a question for you about how you think about the primary care provider and primary care team into this conversation. You know, our healthcare system, you mentioned navigators, but they still have to get into the system, and how do we incorporate them into the conversation?
Right, that's a really good question. And a lot of my research now is in federally qualified health centers, where we are teaching the entire practice from the receptionist to the provider how to implement cancer screening in their routine care. So we have the study, as Dorothy mentioned, on colon cancer screening, and we have our PO1, where we're actually doing 3 things. We're trying to teach them how to do smoking cessation referral, cervical screening, and HPV vaccination. And the problem is, from what I've seen these years and working with these wonderful small clinics and larger clinics, is, you know, when you get trained to be a doctor, nobody teaches you how to implement like cancer screening or a change in guidelines. So we went from 50 to 45 for colon screening. Nobody teaches you how to do that in routine practice. So the first provider we went to was in Quaker City, and she said, you know, I really want to provide the best care I can for my patients. She lives in the community. She knows the community residents come to her. But she says, but I have 10 minutes to talk to my patient, and they come in for, you know, an illness. How am I supposed to talk to them about their sinus infection or diabetes? And for colon screening, I have to tell them what it is, why they're at risk, what the 3 options are. I can't do that in 10 minutes. So what we do is we come in and we teach them, and we give them evidence-based interventions that are tailored to their situation, their barriers, and facilitators to help them. And we teach them how to navigate their patients. So if a clinic can decide, well, this nurse is going to be the navigator, we could teach them how to navigate and not take up their entire day. Because we found evidence-based intervention, which was navigate to fit, mailed fit. We don't have time. Okay, no problem. But then when they got in and saw how many patients it was that they would have to talk to every week, they said, oh, we can do that. And then we taught the nurse who was calling the patients how to navigate their patient. And you know what? You get people to do fits who have never been screened before, and what are you going to find? Positives. And they navigated this one patient to a colonoscopy, and the nurse called us up on a Friday afternoon and told my project manager, Jill, you're not going to believe what happened. Said that patient they navigated to get a colonoscopy had 10 polyps removed. And they knew what that meant, because that's how I always start my presentation to them. So I think we need to listen to the practice and what their needs are, and we need to see their structure. Do we need to train somebody, or can we tell you what a navigator is, teach you how to reimburse for navigation if that CMS rule goes into effect in January? But they need to see the importance of it and what it will do for their clinic. And one last little bit on that. When we finished validating patient navigation in the Patient Navigation Research Program, I asked the James Cancer Hospital to pay the salary, continue paying the salary for one navigator in the breast center. The breast center said, oh, our patients don't need a navigator. It was soon after that the navigator said, I am so inundated. I can only do the uninsured and minority patients. We now have clinicians asking if they can please have a navigator because they've seen the impact. And the latest is the impact on clinical trial accrual. Can we please have a navigator in CLL clinic? Can we please have a navigator here? So I think once you get in and do it appropriately, the need will be there and people will jump on it. Sorry for the long answer. It was a complicated question. Okay, well, thank you all for your attention, and continue with your good work. Thank you.
Don't leave quite yet. Come on. All right, yep, Dr. Yee, I think you have something special for us real quick.
Thank you, Dr. Pasquette. As I said, no adjectival descriptors describe that fabulous and fantastic talk, pointing the right direction, way to go. I want to thank you for a really tremendous B.J. Kennedy lectureship. Thank you for coming today.
Thank you very much.
Okay. Thanks.
Thank you.
Thank you. Sorry for the delay, and I'd like to thank the organizers for giving me the opportunity to present to you today on the work we're doing for the 10,000 Family Study. I'd just like to start by acknowledging the large team that's been involved in this study. I'm here presenting today, but this really is a group effort. We have a large group of collaborators who have worked together on this study, and it really is a great example of team science. In addition to our co-investigators on faculty at the University of Minnesota, we have 3 full-time staff who work on the 10,000 Family Study and a group of additional staff, plus a large number of students and trainees who have been instrumental in helping us get this project off the ground. We're also collaborating with colleagues at the Minnesota Department of Health and also Duke University. And we have some really important community partners who have been instrumental in helping us with this work. So I'd like to acknowledge all of them before I begin with the presentation. So the 10,000 Family Study is a cohort study. So this is a type of study we like to consider in epidemiology as the gold standard for understanding etiology of cancer and other diseases, where we take a large group of healthy individuals and follow them over time to understand which risk factors lead to developing cancer and other health outcomes. So a number of years ago, 5 or 6 years ago, we started talking about how we really needed a new cancer cohort or a new cohort study in Minnesota to study risk factors for chronic diseases. And there were a number of reasons that we thought this was really important. One, that there are a large number of existing cohorts that have been conducted, but they're aging in terms both of the participants in the study and also the methodology that was used. We've really had a lot of advances in technology that can help us improve our understanding of developing diseases. There also are a lot of emerging risk factors that weren't captured in these previous studies. And we were also really interested in using a family-based design to try to help us understand the contribution of shared genetics and environmental factors and how that influences health and disease. We also have a number of unique populations in Minnesota that we also thought deserved to be represented in these types of studies. So before we had funding from NIH, we really had a lot of internal institutional support to develop the infrastructure for this project that then allowed us to transition into an NCI-funded study. One big supporter was the Masonic Cancer Center, who provided the pilot funding both through internal grants and also from the Mason's Gift. We also had a grant from the Office of the Vice President for Research to do a community engagement project that also helped us to develop the project, and then eventually transitioned into NCI funding. And this is a collaborative effort from faculty members all within the Masonic Cancer Center, representing the Division of Biostatistics and Epidemiology and Community Health in the School of Public Health and several departments within the medical school. So this slide just highlights the timeline and the length of time it took us to get this project off the ground. We started discussing the need for this new cohort back in 2015. This emerged from a faculty lunch. We meet weekly to have lunch together, and started talking about how this project would be really great and what could we do to get this type of cohort going. We then started developing some infrastructure and getting ready to do a pilot study. We were fortunate to be awarded a pre-R01 grant from the Cancer Center in 2017 that allowed us to launch the pilot phase of the study. We recruited the first participants for the 10,000 family study at the Minnesota State Fair in 2017. We then continued to build our infrastructure over the next several years, and in 2019, we received a large gift from the Mason's Gift from the Cancer Center to continue to build out this cohort. And at that point, we were finally able to hire some full-time staff to work on the project. We then received a grant in 2020 to do a community engagement project with the Office of Family Development and the Office of Extension at the university that helped us to build some community partners. We launched our community advisory board as part of that project in 2021. And then in 2021, we received a grant from the National Cancer Institute that, where we transitioned to looking at environmental exposures and risk of cancer. So we've continued to build out the cohort over the last 2 years, and just recently received another large award from NCI to continue the study. And I'd just like to spend a minute describing the program that we're funded on through NCI. It's the Cohorts for Environmental Exposures and Cancer Risks, or CECR for short. So in the end of 2020, the National Cancer Institute announced that they would be funding new cohorts to study environmental exposures and cancer risk. And with the infrastructure that we had built with the funding from the Masonic Cancer Center, we were in a great position to apply for this award because we already had everything set up, and we had some pilot data to show that we could recruit this type of cohort. So this is funded under a UG3 UH3 mechanism. So we had 2 years of funding in the UG3 phase to show NCI that we could do the work, and then the opportunity to transition into the UH3 phase to really build out the study. So as part of this mechanism, NCI funded 5 cohorts in the UG3 phase. 3 of us have recently transitioned to UH3 phase, and 1 additional will hopefully be transitioning soon. In terms of our objectives for this project, we really use the catchment analysis that we've done as part of our cancer center to understand burdens of cancer in Minnesota and what type of cohort would make sense for us to conduct here. So our outcome of interest is hematologic malignancy, and this is partly because, as Dr. Yee mentioned, we have higher rates of leukemia and lymphoma in Minnesota compared to the US overall. For those of you who have experience doing cohort studies, you might wonder how we're possibly going to look at a rare cancer like hematologic malignancy in a cohort study. And to be able to do that, we are using a proxy or a biomarker as our outcome for the early phases of the study. So we're looking at clonal hematopoiesis, which is a more prevalent than cancer, but is a very strong predictor of who goes on to develop leukemia and lymphoma. In terms of exposures, we looked at exposures that were relevant in the Minnesota population, including radon, the per and polyfluorinated fluoroalkylated substances, or PFAS, that we've heard a lot about recently, and also glyphosate, which is the active ingredient in the herbicide Roundup. Our study design is a family-based cohort, so we'll be looking at short-term outcomes like cancer risk in older generations, and then we also have the opportunity to look at early life exposures in children. As I mentioned, we have a number of community partners who have been instrumental in helping us develop this study. The first is the Extension Department of Family Health and Wellbeing. They have helped us make a lot of connections in rural Minnesota and also been very instrumental in implementing our Community Advisory Board and helping us with our community engagement initiative that was sponsored by OVPR. We're also working with the SARE, which stands for Hispanic Advocacy and Community Empowerment Through Research. They have been helping us work in the Hispanic community in terms of doing listening sessions, support for participants, and also working with us at events. And then we also have a very large and active community advisory board that we launched in 2021 that were also helpful in figuring out how to implement some of these environmental exposure testing, and also working with us on consent to make sure that participants understand our consent process and what they're signing up for. So this slide just shows an example of some of our community engagement events that we've been working on for this study. We do events in the community where we go out either with our partners or just with staff from the study to talk with community members and also do recruitment into the study. We have connections with a number of professional organizations that serve different communities in the state of Minnesota. We've done listening sessions as part of the cohort grant. We worked with 8 community groups to get perspectives on different communities on what their concerns were related to environmental exposures, and to understand how we could help them participate in a study such as the 10,000 Family Study. And then we also have done a lot of outreach over digital sources, doing education sessions on Facebook and other activities. So in order to participate in the 10,000 Family Study, this is all online to sign up for the study and the initial steps. So we have a screener that people do on an iPad. It takes about 5 to 10 minutes to determine if they're eligible to participate in the study. We then ask people to invite their family members to participate in the study by sending them a link that they can forward on to a family member. Once we have at least 2 people from a family who have completed a screener, the family is then eligible to participate, and they receive links to complete an online questionnaire asking about their residential exposures and also other health behaviors. We invite people to come in for an in-person visit where we collect biospecimens and do some other environmental testing. And then we follow up with people about every 2 years with follow-up surveys to find out how they're doing. So our goal as part of the Seeker program is to recruit just under 9,000 participants for the study. This slide shows how we're doing currently. Currently, we have just under 2,400 participants from about 850 families who are enrolled in the study. In terms of our demographics for our study, we've been working really hard to include rural participants in this study because rural areas typically have been excluded from these types of studies in the past. Currently, we have about 20% of our study population from rural areas, which pretty closely represents the demographics of the state. We've also been working to recruit racial and ethnic minority groups who aren't typically represented in cohorts, and we are working on building relationships, but still have some work to go there to improve our numbers in that area. So I'm going to spend the last few minutes just giving you some preliminary results on our environmental exposure analysis that we've conducted to date. And before I do that, I thought I'd go over our aims for the UG3 phase. So we did have some scientific aims for the UG3 phase of the study in addition to establishing the cohort. These were to evaluate some innovative exposure assessment methods for measuring PFAS and glyphosate, We were hoping we could find ways to do this analysis that wouldn't require us to collect blood and urine samples, because that's difficult on a large scale. And then our other aim was to describe the burden of exposures in the participants in our study. So I mentioned earlier our 3 exposures we're interested in are radon, PFAS, and glyphosate. For radon, we're working with a company called AirCheck. They partner with the Department of Health to offer discounted radon tests to individuals living in the state of Minnesota, and we've partnered with them to send out test kits to participants in the study. Since we want to measure these exposures when the house is closed up, we've conducted our testing in the winter, so we did our first round last year from January to March. For PFAS, we're doing serum measurements, working with colleagues at the Minnesota Department of Health. We also pilot tested measuring PFAS in hair, because we thought that might be an easier sample to collect. But as of right now, we haven't been able to get that off the ground. And then we are measuring the PFAS precursors using silicone wristbands that we ask study participants to wear. So they're just the cheap little silicone wristbands. You ask people to wear them for a week, and then you can get a picture of what types of environmental chemicals they've been exposed to. So that's a third way to measure PFAS. And then for glyphosate, the gold standard is to use measurements from urine samples, and that's also done in collaboration with our colleagues at the Minnesota Department of Health. So far we've completed study visits with just under 700 study participants. But the data I'm going to show you on our results are from a subset of those that we did testing on last winter. So for our first year of radon testing, we completed 238 tests last winter. So we know that in Minnesota, about 40% of homes have radon levels that are above the standard where they should be mitigated. So we did a first short-term test, and then we worked with the testing company and the Department of Health to put together an infographic to help participants understand what their results meant that was a little bit easier to understand than what's typically sent out. So I'm showing you the example we sent people on the left side of the slide. A short-term radon test is really a first screening test that then requires follow-up before you'd make decisions on what to do in your house. So if levels are below 2, it's considered good and no further action is needed. If the levels are between 2 and 8, people are supposed to retest with a long-term test that will give a more accurate measurement. And then if the levels are higher than 8, this is really concerning, and we don't want people to wait for a long-term test. They should do another short-term test. So this was the advice that we gave to participants, and you can see from the graph on the right, we had a relatively large percentage of households that were in the level that needed some further action. We also have measured PFAS using both our wristbands and also the serum samples. This slide is showing some of the common PFAS that are measured, that were detected commonly in our samples. So to date, we've measured 315 samples. We analyzed 24 different PFAS analytes. 8 of them were not detected in any of our samples. 7 were detected in fewer than 10% of participants, and 9 were commonly detected. The 4 that I'm showing you here have been measured by MDH and other analyses in other populations. So if you look on the bar graph, the yellow color represents 10,000 family study participants. There's a black bar that's hard to see that's NHANES, and then the other 2 levels are from work that MDH has done in the East Metro, where they know there's water contamination by PFAS, looking at long-term residents in the gray bar and more recent residents in the white bars. So we noted that in this analysis, the levels that we saw in the 10,000 family study participants were somewhat lower even than the NHANES samples, and much lower than the East Metro levels that had been measured previously. We also were able to look at PFAS in the wristband samples that we had collected. This was in collaboration with our colleague at Duke University. And I'll note that these are not the same analytes that we're looking at in the serum. These are precursors to PFAS, so we're looking at a slightly different outcome measure here. We had 343 samples that we evaluated. Here we measured 13 analytes. 2 were not detected, 7 were infrequently detected, and then we had 3 that we saw that were commonly detected in our population. And we compared our results from 10,000 families to a study in Michigan, and unfortunately the black bars don't show up very well on the slides. But we saw that for the first analyte, we saw much higher levels in the 10,000 family study compared to what was seen in a previous study in Michigan. But for the last analyte, that was not the case. It was much higher in Michigan. So we're still hoping to gather more data to understand what these results mean. And we also, these precursors were measured on the same individuals with the serum measurements, so we also plan to look at the correlation between the two types of PFAS measurements to see if they are comparable using the different methodologies. And then we also have done some analyses of glyphosate in the urine samples that we collected. We measured just over 400 samples for this assay. We found glyphosate detected in almost half of the samples, and the range of exposures was pretty broad. Here I'm showing you some results from 10,000 family study compared to 2 different time points from the NHANES study, showing pretty comparable levels of glyphosate in our study population compared to what's seen in NHANES. So we're doing a lot of work now to ramp up our recruitment to meet our study goals over the next 4 years so we can better answer the relevance of these exposures on cancer risk. We'll also continue to work on our environmental exposures from the UG3 phase, integrating the different biomarkers that or exposures that we've measured and comparing them with data from our questionnaire. We're also interested in looking at how these exposures are shared within family members. So we'll be doing those analyses over the next few months. We've been working really hard with our study team and our community partners to figure out how to report this information back to the participants. We think that it's really important not just to collect these data, but also to share them with participants so that they can take action to reduce their exposures. And we'll be doing additional rounds of radon testing in the upcoming winter season. And I just want to put a plug out. This was a really quick overview, but we do have a poster that will be presented this afternoon in session number 3. So if you'd like more information about the study, you can either stop by our poster or else we have a booth over on the side over here. You can stop by and talk to our staff. I'd like to thank you for your attention, and I'm happy to take any questions.
Thank you so much, Dr. Pointer. So we've got time for one question. We're running a bit behind, so if you've got a good one, put your arm up. What do we got? There we go.
That's very interesting. I guess my question is about the radon, and for the families that had kind of the intermediate or high, are you guys monitoring whether they're being retested, or is the 10,000 family study, sending more kits to them? How is that being addressed?
Yeah, that's a big issue we've talked about. We don't have resources to do the follow-up testing, but we have worked with participants. So in addition to the reports that we've sent back to them, we worked with the Department of Health to put on education sessions that we invited people to talk about what to do next, and they could ask questions about their results. And it's something we're actively working on. Ideally, we'd like to have funding to be able to help them do the mitigation, but that was not allowed with the funding mechanism that we have. So we've been looking for resources that are available in the state and sharing that information with participants, and hopefully moving forward to maybe work with legislators or policy to help get some more resources for participants.
Cool. Well, thank you for the question. Thank you very much for your presentation. And next up, we've got Dr. Dorothy Hatsukami. The floor is yours.
Okay, let's see. Okay, well, thank you. Oops, doesn't look like it's on the screen. I've been asked to talk about transdisciplinary translational science, and I'm a professor of psychiatry and behavioral sciences, and I currently hold the position of being the associate director for cancer prevention and control. So what I've been asked to talk about is how our transdisciplinary team was formed, as well as some of the successes from our team science, and finally, resources provided by the Masonic Cancer Center. So it all started when a colleague of ours, Dr. Harry Lando, had suggested that Steve Hecht and I get together because there might be some mutual areas of interest. And for most of you, you probably know that Professor Steve Hecht is in the chemoprevention and carcinogenesis and chemoprevention program, and his area of focus, he's a chemist, and his area of focus has been to identify harmful constituents in tobacco products, to develop biomarkers that assess exposure to these harmful constituents, and tobacco-caused carcinogenesis. Now, I'm a behavioral scientist, and my area of research has been in the area of characterizing nicotine addiction and finding pharmacological treatments for this addiction. So you would wonder, well, how would we have mutual interests? Well, during the time that we were having conversations, the tobacco industry was starting to aggressively market products that were purportedly reduced in harm. So what they were coming up with are cigarettes that were reduced in carcinogen levels. They were also coming up with products that were heated and not burned, so you don't have the combustion products. They were also advertising smokers to switch from smoking to a non-combusted smokeless tobacco product. And so Steve and I are thinking, are these really effective approaches in reducing tobacco harm among those participants that are using, that are smoking? And we also thought that, at that time, about 40 million smokers were in the US, and many of them were either unable to quit, they tried to quit, but they relapsed to smoking, or they didn't want to quit. And is there a way to reduce the mortality and morbidity among these smokers? So fortuitously, there was an RFA that was issued by NIH for something called tobacco or transdisciplinary tobacco use research centers. So it was a P50 essentially. And the NIH was really interested in trying to see whether people from different disciplines can get together and develop innovative approaches to doing science as well as coming up with innovative solutions. So we decided that we wanted to focus on tobacco harm reduction. And what we did is we utilized some of Steve's expertise, as well as the strengths here at the Masonic Cancer Center, and they included biomarkers, essentially, so we proposed biomarker core, and that included people like Sharon Murphy, as well as Steve Carmelo was very instrumental in doing some of these analysis. And then also, we were very strong in biostatistics, and so we proposed a biostatistics core, and Chapley had led that particular core. We also proposed initially, for our first P50, we proposed 4 projects. And one of the projects dealt with the animal model, and Paul Pentel was leading that project. And it looked at how different doses of nicotine can affect nicotine self-administration in rats. My project involved looking at different approaches to reducing exposure to toxicants in humans, and And the products that we were looking at, well, actually, one of the things that we were looking at was, can we reduce the level of consumption of cigarettes so that the levels of exposures would be low? And what we did is we put 3 patches, nicotine patches, on smokers to see to what extent we can reduce the levels of toxicant exposure. We also looked at the cigarettes that purportedly had reduced carcinogens in them, and we also examined what would happen if smokers were switched to smokeless tobacco products. Ann Joseph led a project looking at if you can reduce the level of cigarette consumption among those patients that had cardiovascular disease, and to see whether that reduction led to significant reduction in exposure levels. And then Debbie Henriquez was interested in looking at reducing secondhand exposure, environmental exposure among kids. So that was the first P50 that we got. In our renewal, we actually replaced Debbie Henriquez with Tim Church, who was interested in looking at whether total NNL, which is a biomarker for NNK, a potent lung carcinogen, whether the levels of total NNL has anything to do with the risk of lung cancer. And we were interested in that question because if we did see a reduction in total NNL, does that mean anything in terms of reduction in cancer risk? So, like I said, we had 2 series of the teachers, and then they sunsetted the program. But one of the major advantages of having a P grant is that it actually leads to a number of other projects as well. So, for example, Irina Stepanov was very interested in smokeless tobacco products and the toxicant levels in these smokeless tobacco products among the products that are sold here in the US. Then eventually she went international and has looked at products that are sold in Africa as well as in India. And what she found is that there is a great deal of variability in terms of the levels of carcinogens in the products sold here in the US. And so subsequently, we did a study looking at how this translates to exposure levels. And what we found is that there is a relationship, even though you take into account the frequency of which the product is used, as well as the amount of use. So what that indicates is that having these toxicants in these products is not a good thing, and there should be a reduction in these toxicants. Dr. Hecht went on to take a look at the impact of electronic cigarettes on the formation of DNA addicts. And what he has found in this study is that DNA addicts are highest among those who are cigarette smokers, lowest among non-users, but we also see higher levels among electronic cigarette users. Now, the electronic cigarette users, it's lower than the combusted product users, like the cigarettes, but higher than the non-users. So electronic cigarettes obviously are not safe. If you take a look at the bottom, Ann Joseph and Stephen Fu, they went on and did a study, an RON, that looked at longitudinal care for smoking cessation, meaning that you don't stop when a person says, well, I haven't been successful in terms of quitting smoking. You just continue to see the patient for a longer period of time to get them to quit smoking. So it was a little bit different approach, the longitudinal care, than what had existed. Typically people gave, for example, nicotine patches for 12 weeks, and if you didn't quit, well, too bad. This is one where they continue to provide care. The projects also led, the T-Turk served as a platform for a couple of other cooperative agreements as well as P projects. One cooperative agreement was focused on what the impact would be of reducing nicotine content in cigarettes. The whole idea was that perhaps we need to have regulation of all cigarettes sold in the US to reduce their levels of nicotine. The other the cooperative agreement and P project involved looking at different types of products, looking at the toxicity, the abuse potential, as well as the consumer perception of products such as electronic cigarettes, smokeless tobacco, and then we also became interested in looking at what happens when you ban filter ventilation. Filter ventilation is when you have holes in the filter to make the smoke a lot more smoother. And so, it adds to the appeal of cigarettes, and people assume that because of the smoother smoke, it tends to be less harmful. So anyway, that was in collaboration with Peter Shields from the Ohio State University Cancer Center, and then the collaboration on the reduced nicotine content cigarettes is with Eric Donnie, who now is at Wake Forest. And what I want to do is concentrate on the Center for Evaluating Nicotine in Cigarettes, which we call CNIC, primarily because it has had the greatest impact, or has the potential for the greatest impact in terms of reducing cancer-related morbidity, particularly lung cancer, but many other cancers as well. And we also developed collaborations with 8 other sites within the U.S. So before I really get into some of the results from the study, what I want to do is I wanted to concentrate or give you a little bit of a narrative in terms of what happened that led us to get to the point where we wanted to actually propose a grant looking at reducing levels of nicotine content in cigarettes. Because what it points out to is that people like us, individuals, might actually have an impact in terms of creating funding resources with a sprinkling of a little bit of luck. So how it started is that the Robert Wood Johnson Foundation and NIH was really interested in this whole concept of tobacco harm reduction. So they gave us money, or the teacher money, to actually convene a series of meetings to see whether we can develop a consensus on the area of tobacco harm reduction, because it was very controversial. Some people said, no, we should not even think about tobacco harm reduction. We just want to eliminate the use of nicotine products altogether. Whereas other people say, hey, look, there's all these people that really can't quit or don't want to quit, and we still need to take a look at those individuals and find ways to reduce their harm. So my co-organizer was Mitch Zeller, who actually had worked under David Kessler, who was the FDA commissioner in the 1990s, essentially. And David Kessler was one of the individuals that said that FDA should have regulation over all tobacco products, which actually didn't survive in court during the time that he was the FDA commissioner. Anyway, we got together and we convened a meeting that included a number of different constituents, I guess. They were scientists that had different opinions on tobacco harm reduction. They were representatives from a number of governmental agencies that included the FDA, the FTC, CDC, as well as NIH. And we also included tobacco control advocates. And through a series of meetings and dialogues, we did come up with the consensus that there is, in fact, a continuum of risk of tobacco products. That you have the combusted products, like cigarettes, that lead to the highest risk to health. And then all the way down the continuum are the nicotine replacement products, the products that have been approved by the FDA to help people quit smoking. In between that are the non-combusted tobacco products, such as the heated tobacco products that I had referred to that don't combust tobacco, and also smokeless tobacco products. So we came to the consensus there is a continuum of risk, but the primary goal, that is the optimal goal, is to get smokers off of all tobacco products, all nicotine products. That should be the primary goal. But we also said that if there are people that can't quit or don't want to quit, then shifting them down the continuum of risk might reduce their mortality and morbidity related to tobacco smoke. So as part of this meeting, the idea also came up that we should reduce the levels of nicotine in cigarettes because that would facilitate the transition down the continuum of risk. And in fact, that concept was not new. It was introduced in 1994 by a couple of colleagues of mine, Neil Benowitz and Jack Henningfield, that said, yes, all cigarettes sold in the US should be below addictive levels, essentially. And so what that would do is that would prevent adolescents or youth to become addicted to cigarettes, and it also would facilitate smoking cessation. And in our teacher days, we did, in fact, do a study that showed that if you do reduce the levels of nicotine in cigarettes, you can reduce cigarette consumption. You can also reduce the levels of toxicant exposure as well as dependence on cigarettes. So it seemed ripe to revisit this idea, and in fact, Mitch Zeller and I decided to convene a series of meetings, again with these scientists, people from the different governmental organizations and our agencies and tobacco control advocates to discuss whether the existing science, it tended to be minimal because there was no movement towards developing this area of science because at that time there was no tobacco, there was no agency that had regulations over tobacco products. So we had limited amount of research, but we wanted to take a look and see whether this might be a feasible approach to regulate the levels of nicotine in cigarettes. And the people that were at this meeting said, yeah, it sounds great. You know, we should move forward with this approach, but there's a lot of gaps in the research. And so we convened another series of meetings that we decided to develop this research plan, you know, in terms of what we need to know in order to move this regulation forward. And as part of that, we, the National Institute on Drug Abuse, decided to provide investigators cigarettes that varied in doses of nicotine. So that was really, really exciting. So what happened in 2009 is that there was a Tobacco Control Act that was passed that did, in fact, give the FDA jurisdiction over tobacco products. And as part of that law, it stated that the agency can reduce the levels of nicotine in cigarettes to be minimally addictive just as long as it's not zero. Now, who knows why they said not zero? In part, it could be due to the fact that the law also states that the FDA cannot ban any categories of tobacco products. So they cannot ban smokeless tobacco, they cannot ban cigarettes, they cannot ban other tobacco products. So that may in part have been the reason, because if you do put it to zero, then you're essentially banning the cigarettes. So that came into effect 2009. Along with that came a lot of money that the FDA had acquired, primarily through user fees that cigarette manufacturers or tobacco manufacturers were paying to the FDA to evaluate tobacco products. But as part of that, they partnered with NIH so they can use some of the money to fund research. And lo and behold, we were there with a plan, in terms of reducing levels of nicotine content in cigarettes. And lucky for us, many of the people that we had in our meetings that were representative of different government agencies, such as the CDC, the NCI, even NIDA, they went to the FDA to assume leadership roles. So what that meant is that we had this whole group of people that were very supportive of this idea of reducing levels of nicotine in cigarettes. So we did get a huge grant and a cooperative agreement, and what we demonstrated is that indeed we identified a threshold dose for nicotine that we believed was minimally addictive. At this dose, we saw indeed a reduction in cigarettes. We saw a reduction in dependence, toxic exposure, and an increase in quit attempts. And we found this not only in a general population of smokers, but smokers that had psychiatric disorders such as schizoaffective disorders or depression. We also found a reduction in smoking among youth as well. And then some of our colleagues also got grants, and they found that among people that are of lower SES, among people that have substance use disorders, you also saw a reduction in smoking at the specific threshold dose of nicotine. So we went on and said, okay, so now we've identified this low dose of nicotine that will lead to a reduction in dependence. What's going to be the best approach? Should it be an immediate reduction, on a set date, all cigarettes should be at this particular level, or should it be a step-down approach? So again, we did this huge clinical trial, and we found that, as you would expect, the immediate reduction is what led to a greater reduction in dependence, cigarette smoked, toxicant exposure, and in fact, it led to greater increase in terms of cessation rate. So we propose that there should be an immediate reduction in nicotine. Well, because there's been such a big body of scientific evidence that was created, the FDA decided that, hey, this might be a really good approach to to improve public health. And so what they did is they issued something called Advance Notice of Proposed Rulemaking. And so this is one in which the FDA says, hey, we're thinking about this idea, and we want people, the public, to comment on it. We want to know what you think of it. And the FDA, like I said, decided to move forward on seeing what the public reaction should be, primarily because of the scientific data that was building. And also they did a modeling that showed that if there was a reduction in levels of nicotine in cigarettes, then 8 million tobacco-caused deaths would be averted by the year of 2100. 8 million. I mean, that's really huge. So anyway, they did propose, they did let the public know, and the public, like scientists and obviously the tobacco industry had put in a lot of comments, and then it was quiet for a while. We didn't hear anything from the FDA. We didn't know whether they were really interested in continuing to pursue this idea or not. So we went on, we decided to renew our Scenic, and so Scenic 2.0 occurred, and that was focused on looking at what would happen if we reduce the levels of nicotine content in cigarettes in the context of alternative nicotine delivery systems that were non-combusted. So in the context of having the availability of electronic cigarettes, something called nicotine pouches, which just contain nicotine. They look like smokeless tobacco products, and nicotine replacement therapies. And we did this because in the prior studies, participants could only use the assigned cigarette products, so they can only use very low nicotine content cigarettes. So we wanted to know what would happen if people had access to a marketplace that had all these other products that were in the market. And so what we found is that indeed people who were assigned to the very low nicotine content cigarette marketplace, and we actually developed an experimental marketplace that looked like an online shopping, and they can pick their products that they want. We gave them points, and they can exchange these points, which were valued $1 to, for various products. And if they didn't want to spend it on products, they can exchange it for cash at the end of the study. But anyway, so what we found is that indeed, even though the people with a very low nicotine content condition were exposed to all these alternative products, we saw a decrease again in cigarettes dependence. And we also saw an increase in terms of uptake of these alternative products over time. And what happened is they shifted from using both cigarettes and these alternative products, but then exclusively using these alternative products that tended to result in less toxic exposure. So, it was a good thing. It wasn't as though the value, the public health benefit of a reduced nicotine content cigarette would be compromised in the context of all these other alternative products. So recently, the Center for Tobacco Products director had announced that they are, in fact, moving forward to propose a rule to reduce levels of nicotine content in cigarettes. So we're really excited about that. They were thinking that it would happen in the fall. It hasn't happened. They're thinking maybe in the winter. I'm not sure, but it likely will happen next year. But the one thing I learned is that the tobacco industry will come back and they will sue the pants out of the FDA because, this, again, is a billion-dollar industry here, the cigarettes are. And I also learned that the administration really the White House administration does make a difference. So I'm not sure whether this proposed rule will, in fact, occur during my career, but I am very hopeful. So what I hoped I had illustrated is, what could happen when you do have disciplinary translational pipeline of research. And so, here we had a collaboration of people that were interested in looking at the harmful constituents of tobacco products, determining their harm by doing in vivo, in vitro research, developing biomarkers to assess their harms, conducting human clinical trials, and currently we've been doing some epidemiological studies, partnering some of what we've learned in terms of the harmful constituents and merging them with some national survey data, and so utilizing these epidemiological surveys to help in terms of developing policies. And these policies obviously have to have an equity focus. And so, that certainly has led to the proposed rulemaking, which we hope will occur next year, for reducing levels of nicotine in cigarettes. But I also want to mention that it has led to an advanced proposed rulemaking where the FDA said, hey, we're thinking about reducing the tobacco-specific nitrosamine NNL in smokeless tobacco products. Unfortunately, they haven't moved on this issue. Well, we'll see whether they will in the years to come. And locally, it has helped in terms of continuing the smoke-free rules or regulations here in Minnesota. What we did was we looked at the urines in people, the hospitality workers, before we had a smoke-free law and after. And what we found is that the levels of carcinogens in the hospitality workers were really quite low, well, non-existent after the law was passed, and that led to the continued support of this the smoke-free law. So what I want to just end it on is to say that we did get tremendous support from the Masonic Cancer Center. We utilized the analytic biochemistry shared resource that includes people like Steve and Sharon and Irina Stepanov. The biostatistics shared resource that included not only Chapley but Joe Koopmeiners and Shanghua Luo, And then space was provided to us at 717. We also received some pilot funding from the Cancer Center for our P grants and cooperative agreements. And importantly, we had incredibly supportive Cancer Center directors, and they started off with John Kersey, and then, of course, Dougie. So thank you so much for your attention, and I hope I've demonstrated to you how transdisciplinary translational research can really bear a lot of fruit. Thank you.
Awesome, nice job. Thank you very much, Dr. Hatsukomi. So we've got one more surprise from Dr. Yi. Here he comes.
Oh, my.
Thank you. I guess we're going to cut back on questions given time, Dorothy, so please feel free to capture Dr. Hatsukami anytime to ask a question. So one of my favorite awards to give out at this is the Mentor of the Year Award. As you know, when we plan this meeting, we solicit all the trainees to nominate a Mentor of the Year. It is always highly competitive. The committee who makes the decision has a tough time. And I'm pleased to announce that this year's Mentor of the Year is Dr. Carol Lang. I think you know Dr. Lang, or if you don't, I had the good fortune when I came during the last century to recruit Dr. Lang as a partner. She's risen through the academic and national recognition ranks. She's mentored, I don't know how many students, Carol, and postdocs, 30-ish or so. Yeah, and has done a tremendous job bringing young trainees forward. And I always think the term mentor is probably, you know, you got this hierarchical description, but I think she's been a mentor for me in my work. She's helped me understand some of the things I didn't understand, so I'm thrilled to announce this, to present this award to Dr. Lang.
Up next is the most popular parts of the day, the faculty flash talks. Each of the 6 program areas are nominated a faculty member to give a 3-minute presentation that you get to vote onto. To vote, text the number 1 through 6 to the number that will be on the screen at the end. Those instructions will be on the screen so that you can just follow that and make sure you cast your vote. The winning speakers program will get to host an external speaker for the 2022-23 Masonic Cancer Center Seminar Series, and the winner gets $5,000 and Masonic Cancer Center Shared Resource Services. So up first is Dr. Mark DeStefano. I screwed it up even though I asked the guy. All right. So presenting on the behalf of the carcinogenesis and chemoprevention program, you've got 3 minutes. Take it away.
All right, thank you so much. So good afternoon. My name is Mark Stefano, and I would like to introduce you to some of the work that we're doing in my research group. So the plasma membrane plays a critical role in living cells. Beyond simply defining the boundaries of the cell, the membrane plays an essential role in the process of signal transduction. Importantly, signaling molecules such as growth factors do not typically enter cells to elicit their effect. Rather, they bind to receptors on the cell surface and use various adapter proteins bound to the inside membrane to transfer information about receptor occupancy to the cellular interior. What holds the adapter proteins to the membrane? One key mechanism involves the use of lipid anchors shown attached to the RAS protein in this figure. That is what my research group works on. The molecule shown here is an inhibitor of the enzyme that links the lipid anchor to RAS protein. The image below indicates how the inhibitor overlaps with part of the RAS protein, thus blocking RAS from binding to the lipidating enzyme. By inhibiting membrane localization of RAS, these molecules effectively shut down uncontrolled growth of cells. Beyond new drugs, chemistry also facilitates the creation of probe molecules that can be used to discover new features of biology that can be exploited for cancer therapy. The probe molecule shown here is an analog of the natural lipid shown above. Cells grown in the presence of the probe incorporate it instead of the natural substrate since it looks so similar. However, the probe contains a carbon-carbon triple bond that does not occur in nature, which allows us to selectively label all of the approximately 100 lipidated proteins in the cell and identify and quantify them in a single proteomic experiment. In this graph, we show how decreasing the level of a chaperone protein that assists in the lipidation process leads to a global decrease in the protein lipidation. Since breast cancer survival correlates with decreased levels of the chaperone protein, these experiments provide a molecular mechanism for how this occurs and suggest a new approach for breast cancer treatment. In the last 10 years, a variety of new cancer therapeutics have been created that are based on antibodies. These include antibody-drug conjugates, where the antibody binds to receptors overexpressed on the surface of cancer cells. Shown here is a simpler alternative that uses a small protein that is easy to produce. The protein has been engineered to bind to cancer cells and chemically modified to deliver a variety of cargoes shown in red. When attached to a toxic molecule, the resulting protein conjugate selectively kills cancer cells. When attached to the appropriate metal ion, it can be used for PET imaging as shown in this paw inflammation model. Given the modular strategy used to assemble this structure, it should be possible to create materials that could be used to both image and treat tumors. Thank you.
All right, up next is Dr. Tu Trong, and he is presenting on behalf of the Cellular Mechanisms Program. Dr. Trong, you're up. You got 3 minutes. All right, to save on time, I think we're going to go on to the next presentation and then come on back. So we will be doing Dr. Hai Dang Nguyen, presenting on behalf of the Genetic Mechanisms Program. All right, doctor, you got 3 minutes.
My name is Hai Dang, and I'm assistant professor in the Department of Pharmacology, and I'm in the Genetic Mechanisms Program. So in our lab, we're very interested in studying this new emerging cancer mutations in RNA splicing factors. As you can see in the top left, there's about 119 splicing factors mutation across all the pan-cancers of 33 different cancer types, and causing a lot of alternative splicing in genes, which we usually don't detect if you do DNA sequencing. And particularly, there are some hotspot mutations that we identified within myelodysplastic syndrome and acute myeloid leukemia, which is in SRSF2, SF3B1, and U2F1. And in MDS, these patients usually have adult bone marrow failure and actually causing red blood cells, loss of red blood cells production. So we're really interested in understanding what are how the splicing mutation drive this pathogenesis, as well as to identify novel targeted therapy. Just to summarize what we've done in the last 3 years, is we have generated multiple different murine models, as well as using human models and patient and primary AML patients, and we identified that across all the different cancer types, that all the splicing mutations are particularly sensitive to FDA-approved PARP inhibitors. And these inhibitors have really been used mainly in breast and ovarian cancer with homologous recombination repair defect. So then we were wondering as to why these blood cancer cells are actually also sensitive to PARP inhibitor, and whether these are actually due to a driver of this homologous recombination repair defect. But to our surprise, what we found is that although all 3 splicing mutations are sensitive to PARP inhibitor, only SF3B1 mutant cells have a defective HR, but the other 2 splicing mutants are not. And just to summarize what we have identified is that these 2 splicing mutations, particularly in U2F1 and SRSF2, actually have this accumulation of a new nucleic acid structures called R-loop, which has an RNA-DNA hybrid in the genome, that the cells really require PARP function to survive. And so with this result, we actually now propose a new clinical utility of using PARP inhibitors in MDS and acute myeloid leukemias to treat with these hotspot mutations that can be identified in patients to sensitize these cancer cells. And what we are doing now in the lab is really building out a pipeline of workflows to do more mechanisms of studies and how these cancers are really driving these mutations, as well as doing biomarker development and also targeted therapy. Thank you.
All right, thank you guys. All right, so our lab studies ER-positive breast cancer, and this subtype accounts for a majority of cases and is defined by the presence of nuclear receptors such as estrogen and progesterone receptor. And so patients with this subtype are treated with endocrine therapies, and they tend to respond very well, but many of them do eventually develop resistance that can progress to metastasis. And so our lab is focused on understanding the mechanisms that lead to therapy resistance. And so there are a lot of things that lead to resistance, and those can include things like mutations and altered protein interactions. So for example, we know that ER can acquire mutations in its ligand binding domain, and these can lead to not so great things in breast cancer, and it's also dependent on influencing factors in the environment as well. We also know that in breast cancer that there can be altered protein-protein interactions, and I think all of us can agree that coming out of the last few years out of COVID, that we've really started to kind of appreciate the interactions that we have with other human beings and the relationships that we form. And so let's take Katie Heron for an example. She just started as a new student at a high school, and she's just trying to figure out what the social climate is. And I think we can all, to some extent, remember how awkward high school interactions can be. So on her first day of school, she meets Janice, Ian, and Damien. And they're good people. They become her friends, but they do encourage her to partake in some behavior that isn't so great. And that's kind of what happens when PR and ER talk to each other in breast cancer. And it's not too long before Katie comes across Regina George and the rest of the plastics and gets caught up in their web of high school drama. And so Regina George is really, really good at persuading people around her and influencing them to do things the way she wants them to and exactly how she wants them to, which is kind of how mutant ER behaves in breast cancer to drive resistance. So what are we doing to help Katie make better choices in life? Well, our lab is focused on identifying novel protein interactions and then using that information to create targeted inhibitors that will block those interactions from occurring. And so we hope by blocking these interactions, we can block any not so great things that happen downstream of that interaction. Because I think we can all agree that we probably weren't the best versions of ourselves when we were 16 and in high school with hormones flying everywhere. And so we're hoping that the inhibitors we generate will help Katie make some better choices in life and give her a chance to self-reflect and grow as a person. Thank you.
Okay, up next is Dr. David Hines, presenting on behalf of the SPECS program. Dr. Hines, you've got 3 minutes. Before he starts, though, I need Dr. Viva Vaziz to come on up here, or to the back, or you're right here? Okay, perfect, you'll be next. Thank you.
So I'm David Haynes, and I'm an assistant professor with the Institute for Health Informatics, and I study health disparities and cancer disparities. Over 17 million people are currently surviving cancer, and this is a traumatic experience, obviously, for them and their family when they learn about this. But every single one of them, when they're given this news, are met with a healthcare professional who will give them a binder full of flyers and pamphlets of community organizations and resources that they can connect with if they need any sort of social need, like food, housing, transportation. When they finally find these binders, all the programs are out of date. Organizations are closed. It's not working. And so they're desperately trying, no one thinks they're going to need this, but they're desperately trying to get access to the resources that they need for them and their families. So what I've done is I've designed a social care referral platform to essentially digitize and revolutionize this whole process. It allows patients to self-screen at their own home about their own social determinants of health, identify the organizations that are out there, and refer themselves directly to those organizations. We have a web portal that allows community-based organizations to update and manage their programs in real time, and we have an EHR HIPAA-protected interface that allows clinical teams to be a part of this process and understand when their patients actually get access to the resources they're looking for, as well as refer their patients to other community organizations that they don't actually know about. So cancer is a unique disease that's time and resource intensive. And because of this and this multi-collaborative sort of interdisciplinary teams, there's often opportunities for people and patients to fall through the cracks and delay care and reduce survivorship. And even though treatments have improved our ability to survive cancer, there's certain populations that are just not reaching these sorts of goals. And so we need to be able to really focus on the social determinants of health and how those are, and understanding how those are impacting their survivorship. In addition to this, our technology allows them to provide timely patient access, the patient's timely access to the resources to increase survivorship. And this is really focusing on our patient navigators and our social oncology workers, or social workers that are embedded with oncology teams, to improve their ability to scale. Because right now, our social workers are really about 100 to 1 in terms of patients, and so we need to be able to improve their ability to impact the patient population. We're currently right now conducting a pilot with the Minnesota Cancer Alliance, which includes about 14 community organizations and 50 patients. You can see some of their needs here, and we're understanding how the community organizations are able to directly provide resources to these patients and how the patients are able to utilize these resources today. I have some students today that will be presenting in the last poster session. Love to talk to you.
All right, up next is Dr. Viva Vajij, presenting on behalf of the immunology program. Dr. Vajij, come on up.
By the time you're 18, you experienced many different common pathogens like cytomegalovirus and like Epstein-Barr virus. Your immune system reacts to these viruses, and T cells, CD8 T cells, are induced, and they populate all your healthy organs and tissues, and they stay for the rest of your life. If those memory T cells just see a little short 9-amino acid peptide that induces them to reactivate, and then they sound the alarm and say, oh my God, I think the infection is back. I need help at this location. Come on, all you immune cells, come over here and help me out. So when you have a cancer or malignancy developing out of your healthy tissues, that tumor that grows out will also contain these antiviral CD8 T cells in that tumor. So we were wondering, can we change the tumor microenvironment by mimicking a viral infection just by sprinkling some of these viral peptides on top of the tumor? So we got some tumors from the OR, we sliced them up, we put them in a dish, sprinkled those peptides in, and we did see that there was an inflammatory signature induced when the viral peptides were seen and triggered those memory CD8 T cells. So then we wanted to know if these memory CD8 T cells that are virus specific can alter tumor growth in vivo. So we had mice in the lab where we infected them with a known virus. We came back later and we gave those mice a tumor, completely antigenically unrelated, the tumor and the virus. When we could palpate the tumor, we injected the tumor directly with peptides and saline from that viral infection, and when this was combined with PD-L1 blockade, we found that most mice cleared the tumor. This works by these CD8 T cells that are in the tumor that are virus specific, responding to that little peptide from the virus. It mimics a viral infection, and that's what the T cell thinks is going on, and again, sends out this alarm and says, help, I need help in this location, and the innate immune system is activated locally, and cells from the circulation are also recruited to the site. What's great about this is we don't care about those pesky cancer neoantigens, which are different within a tumor and between patients. These viral peptides are very common within large groups of people. So now we are getting ready to do a phase 1 clinical trial in people, which we hope is going to start recruiting patients this coming year. We're going to be injecting EBV and CMV peptides in saline directly into tumors in patients, combining that with checkpoint blockade, PD-L1, CTLA-4. Of course, primary outcome is safety. It's a dose escalation study, but we will be looking pre and post in the tumor, doing biopsies to look for changes in the inflammatory state of the tumor and monitoring the patients throughout the entire time. Thank you.
Well, that was awesome. Congratulations to the faculty Flash Talk presenters. That's not easy to do, so thank you very much for doing that. All right, so now it's time to vote. You got to text the number 1 through 6 for the person of which you are voting for. The phone number. Oh, we have one. Where's my cue on it? Okay. Well, my apologies. I cut him off. My apologies. Nick Zorko, who is, who are you, what's your department? TCT. TCT, great. He's talking about TCT, which he knows more than I do about it, and he's got 3 minutes to do that. Sorry about that.
Okay, thank you. It's always hard being the Z. My name is Nick Zorko. I am an assistant professor in HOT, and my focus is developing immune and cellular therapies for treatment of solid tumors. In particular, we're interested in developing natural killer cells for treatment of prostate cancer. I'm going to talk today about enhancing natural killer cell function with androgen receptor blockade, and we'll go through in order in the slides the arrows. You've probably heard a lot about our tri-specific killer engager, or TRIKE platform already. I'm not going to dwell on a lot of this, but this is homegrown natural killer cell engager at the University of Minnesota. So this functions essentially by forming a physical link between the natural killer cell and the tumor target of interest, redirects the NK cell function, and destroys the tumor. My main focus is prostate cancer, and the backbone of prostate cancer therapy is blocking androgen or testosterone. We can do this in several different ways. It can be an injection given every few months, it can be a pill taken daily, or it can also be a surgical castration as well. The other option is to block androgen receptor function itself by using medications called androgen receptor targeted therapies, ARTs, and in this particular example, we're going to focus on the use of a drug called enzalutamide. The important thing about enzalutamide is that it blocks the interaction between androgen and androgen receptor and inhibits the formation of the heterodimer. This heterodimer is what leads to transcriptional activation of the AR program. This also forms a backbone of treatment for men with prostate cancer by blocking androgen receptor. The important thing to think about is that in prostate cancer, all of our patients that are going on trials are required to have a castrate level of testosterone, so less than 50 nanograms per deciliter. None of our other cell therapy trials have this condition or this requirement, as they're almost all leukemias or lymphomas that are not hormone sensitive. So we really don't know what will happen in this context that we've added. Will it enhance an NK cell therapy or T cell therapy? Will it stay the same, or will it inhibit? So that leads us to the next question here. There's a group at Oregon Health Science University, Amy Moran's lab, took a look at this and published early or late last year the effects of androgen receptor blockade using enzalutamide in T cells. They took T cells from normal donors and prostate cancer patients, paired them with enzalutamide and an anti-PD-L1 antibody and PD-L1 positive tumor cells. And in the lower right, we can see highlighted in the red box, there was an enhancement of multiple levels of effect in these T cells, including increased granzyme B production, so cytolytic granules, but also increases in the level of inflammatory cytokines such as tumor necrosis factor. We took this idea the next step. We're focused on natural killer cells, and we wanted to see what happens when we block testosterone with enzalutamide and NK cells. So we took normal donors, added enzalutamide and our TRIKE along with B7H3 positive tumor cells, and what we see in this preliminary data is that there is a dose-dependent effect of enzalutamide. The more enzalutamide we have, the higher effect in degranulation there is in the TRIKEs. So we're really hopeful to repurpose this enzalutamide in addition to our natural killer cell therapies to bump the efficacy. Thank you.
Okay, so we're going to keep on going, and the next session is called Leveraging Immunomodularity Networks to Tackle Cancer with Dr. David Odie. and Dr. Brandon Moriarty. The first will be Dr. Odie, who will present and is the professor of biomedical engineering. The floor is yours.
Okay, good afternoon, everybody. My name is Dave Odie. I'm a professor in the biomedical engineering department, and it's a pleasure to be able to speak to you today. I'll tell you a little bit about the work that we've been doing over the last several years to apply physical sciences to oncology research and develop a cancer bioengineering initiative at the University of Minnesota. So I'll tell you about some of the team building efforts and some of the scientific accomplishments of what we've done so far. So if you think of biomedical engineering, which is my appointment, you might think of devices for cardiovascular or neural applications. And but we thought there's a lot more that engineers could do, particularly in oncology. And of course, engineers have made important contributions to oncology. If you think of imaging, like MR imaging and CT imaging and so forth. But to really delve into the physics of cancer progression, how do cells proliferate, migrate, and how do immune cells try to respond to tumors, these types of questions, the mechanics of all those processes, perhaps engineers could contribute to that understanding. So about a decade ago, a little more than, our Department of Biomedical Engineering, under the leadership of Bob Tranquillo, started to invest in developing cancer bioengineering as a thrust area in our department, and that led to new faculty hires, and key new hires were Paulo Provenzano and David Wood. And that led us to start to think about how we could do collaborative research together around physical sciences and oncology. And at that time, the National Cancer Institute had a U54 call for physical sciences and oncology centers. And we started to strategize about developing such a center, which involves having whiteboards and PowerPoints and sandwiches. And out of that emerged the U54 proposal. And the first try didn't go well, and nor did the second one. But on the third try, we finally succeeded. And the theme that we ended up focusing on was the mechanics of cell migration, that we could maybe develop and experimentally test a computer model for the mechanical dynamics of how cells move through tumor microenvironments, kind of flight simulator, with which we could design therapeutic approaches to potentially disable that mechanical machinery that's driving cell migration and tumor spread. So that's kind of shown schematically here is the framework for our U54. And we broke it down into two projects, one that focused on the machinery inside the cell, cytoskeleton and adhesion. That's project one, led by myself. And the second project focused on the environment that cells exist in, the mechanics and chemistry and architecture of that. That was project two, led by Paulo Provenzano. And we focused on really dismal prognosis cancers such as glioblastoma, and where cell migration is key to spreading and disseminating the disease. So in glioblastoma, despite surgery, chemotherapy, focused radiation therapy, these tumors almost inevitably recur and spread throughout the brain, leading to the demise of the patient. And similarly in pancreatic ductal adenocarcinoma of dismal prognosis, with cell migration being a key driver of progression. So the simulator we developed looks schematically like something like this, where cells are able to get traction in their environment through these green adhesion bonds, like think of integrins, for example, and they do that through making connections between their actin cytoskeleton in red and molecular motors, myosin 2 motors in blue, and that allows them to get traction forces in their environment. And we apply fundamental conservation principles of mass conservation and momentum conservation to try to predict the trajectories of the cells under various conditions, mechanical and chemical or otherwise. And that led to this cell migration simulator, where here you see a single cell being simulated according to this model. And in this particular instance, migrates nicely off as new protrusions are nucleated, migrates nicely off to the right-hand side. And we can change the environment too and see how that influences how well a cell migrates. And what we found is that there's a sweet spot or optimal stiffness predicted by the model, such that at medium stiffness, cells will migrate well, but at very high or very low stiffness, they get poorer grip on their environment and they migrate relatively poorly. So it's biphasic with respect to stiffness in the environment. They've kind of dialed it in or tuned themselves to a specific type of stiffness. And that led us to also want to understand how to test those models in more in vivo-like settings, including using optical imaging where we've used fluorescence confocal microscopy and other modalities in combinations to, in this case, visualize glioblastoma cell migration through mouse brain tissue and their possible association with white matter tracts and blood vasculature as architectural features that they can follow. And furthermore, we wanted to understand whether the cells have this sweet spot type of behavior, whether they're attracted to certain stiffnesses or not. An example of that is shown here, where glioblastoma cells are plated on an alternating stiff, soft environment where 8 kilopascals, the gray region, is kind of their preferred stiffness according to traction force measurements. And working with Mark DiStefano, who you heard from just a moment ago, creating alternating stiffness patterns by photo-cleaving cross-linkers in the underlying hydrogel. We're able to show that these cells, let me start that again. These cells would accumulate over time on these softer regions as predicted by the model, demonstrating the ability to respond to gradients of stiffness in the environment. We found that breast cancer cells also had this ability, and that we could change their ability to go from in the stiffer direction only in the top case here in control cells to cells that were capable of moving towards softer directions and accumulating an intermediate stiffness, shown in the bottom panel here, when adhesion proteins talin-1 and talin-2 were silenced. Again, another prediction of the model. So overall, what we discovered was that cells can respond to gradients of their environment, both towards positive, stiffer, and softer environments. And it's the negative durotaxis that had not been observed previously that we observed for the first time that was predicted by this cell migration simulator. And another application of this model was to understand what is the clutch or the adhesion that cells use to get grip in their environment in brain tissue specifically. And so here, if we had a gene that was controlling how adhesive the environment is, something that makes it sticky or less sticky, then on the right-hand side, a highly adhesive environment would lead to cell protrusions going out and kind of getting stuck in their environment and not being able to let go in any direction and therefore move poorly. Whereas on the left-hand side, protrusions would go out and get pulled back in, thus not allowing the cells to get good grip in any direction, also moving poorly. Only in the middle case do cells get that good balance of getting sufficient grip in the environment, but also letting go, are they able to move in an efficient manner. So that predicts that if we could control a clutch or adhesion molecule in a genetically engineered mouse model, then we would predict that at some intermediate level of that adhesion molecule, perhaps at the wild type background level versus knockout or overexpression, then there would be excellent migration relatively and poor prognosis presumably for the animal. And so working with the late John Olfest, used the Sleeping Beauty mouse model that he and Dave Largett-Spata developed here at Minnesota to induce common oncogenic driver mutations that are correspondent to glioblastoma patients, and tracked the trajectories of individual GFP-labeled glioblastoma cells in mouse brain slices to see if this hypothesis is correct, which would confirm that CD44 is acting as a major clutch. And what we found was that by tracking individual cells, you can see the green tumor cells moving out into the darker brain region here, away from the tumor mass in the lower left, that we could measure those speeds and do that as a function of genetic background, and indeed found that the intermediate level of CD44 had this property of fast migration. And if you went too high or too low, cells did not migrate nearly as efficiently. And that was correspondent to the survival of the mice, which was poorest where the migration was fastest. And we examined human transcriptomic data and found the same U-shaped curve, that in human survival as a function of CD44, survival is poorest at intermediate level of CD44. And with it, by subtypes, this also varied as well. So the proneural subtype was relatively low in CD44 and therefore suboptimal, presumably. And the mesenchymal subtype was relatively optimal, or like maximal migration, not optimal from a patient's point of view, poorest survival. So we've tested that hypothesis, where proneural sits left of this optimum and mesenchymal sits at the optimum, and examining human glioblastoma cell lines from these two cohorts of mesenchymal and proneural subtypes, finding that indeed in the top row, the green glioblastoma cells migrate really efficiently. They also get excellent traction in their environment and are literally tearing up the blood vessels in many cases, whereas in the bottom row, cells are poorly migrating because they get poor adhesion in their environment. So overall, in Project 1, some of our findings were this discovery of optimal traction forces as a function of stiffness and the role of CD44 in mediating adhesivity in the brain, and also the discovery of negative durotaxis as predicted by the model in spatially varying stiffness environments, and then potentially mechanically targetable differences by subtype within glioblastoma. And in Project 2, which was led by Paolo Provenzano, which focused more on the extracellular environment, Paolo's project focused on the stromal environment, how it affects tumors, very commonly associated with mediating growth and progressing to metastasis and frustrating therapeutic interventions. And could we potentially re-engineer the stroma to flip that script and make tumor growth poorer and facilitate therapeutic interventions? And through the course of the U54 grant, made a number of key discoveries, which I won't have time to go through all of them, but just highlight a couple, that by micro-patterning the environment, being able to visualize how cells can create contact guidance by orienting their focal adhesions along these patterned surfaces, and that enables them to have directionality to their migration. And furthermore, using micropatterns to create competing directions of, in one direction, vertically cell substrate type of adhesion, and in the horizontal direction, lines of cell-cell adhesion molecules, E-cadherin in this case, and examine whether cells have a preference to go with cell-cell or cell-substrate adhesion. So another kind of utilization of microfabrication. And another use of microfabrication was to create small tissues. In Dave Wood's lab, they have this microfluidic technology where they can create small droplets to which cells can adhere, and cells can grow on that and create a little micro tissue, and then that can be observed in a multi-well, micro-well custom format to visualize cell invasion as seen in the lower movie here. So altogether, it was great to build this community of physical sciences and oncology. This grant is now ended, but maybe our legacy will be the people who trained here and have gone on to other things now in academia and industry and government laboratories, and we're very proud of the training that they got here and the community that they helped build. And through the process, being a part of a U54, and I think Dorothy talked about this earlier, having a center grant like this enabled us to connect to other entities that are going on around the country. There were 10 physical sciences and oncology centers in the US. We were one of them. And so that allowed us to host the network as a PI meeting in Minneapolis in 2019. And so we invited all the awardees to come to a meeting that we helped organize. And so that helped enhance our visibility, I think, and put cancer bioengineering as happening at Minnesota. We also developed new techniques for modeling that weren't just computer-based or mathematics-based, but human movement-based, where we simulated tumor growth and other aspects of cancer biology using human movement in collaboration with the Black Label Movement Dance Company led by Carl Flink in the Theater Arts Dance Department. And this helps break down communication barriers where maybe not everybody in my collaborative spheres talks the language of mathematics, but at least we can talk about human movement and forces and pushing and pulling and things like that. So that gives us a way to maybe democratize this scientific process and engage not only with people who are less mathematically inclined, but also the patients and their caregivers and clinical teams as well. Another aspect of building a solid team is having an excellent staff to support it. And here we were really fortunate to hire Bo Connolly as our center manager for the PSOC. And for those of you interested in building centers, I'd be happy to share our experience and journey in this, which I'm trying to do today. But some of the key attributes that Bo brought to us were strong experience in project management, kind of keep setting goals and keep moving towards them. Entrepreneur experience, himself being an entrepreneur and having his own startup. developing a strategic plan, and previous experience in clinical trial management. So those were key. He helped us then develop a strategic plan and set goals that we were kept making decisions around based on those goals to guide our decision making and not get too meandering in our approach. And we're really grateful for the University of Minnesota institutional funds from the Cancer Center, from Doug and the leadership team, from the Office of Academic and Clinical Affairs, Tucker Labine and his leadership, medical school, Jacob Tolar and Tim Shacker, as well as the Institute for Engineering and Medicine led by John Bischoff. All of these entities helped contribute discretionary funding that allowed us to grow in new areas. As the PSOC came to an end, that was important as we started to visualize new horizons. We were trying to develop new funding to continue this work, and we invested in immunotherapy, in particular immuno-oncology, and this bore fruit after 3 tries in new PO1 funding to create an immunoengineering translation accelerator. And the way this is structured is 3 projects, where Project 1, led by myself, is focused on brain cancer and modeling. Project 2, led by Paolo Provenzano, again, focused on pancreatic cancer and modeling. And Project 3, T-cell genome engineering, led by Brandon Moriarty and Bo Weber. And to try to integrate these together to intake input therapy concepts, and you heard about peptide alarm therapy just a few minutes ago, and try to put them through these various engineering technologies to optimize and de-risk them, and hopefully then accelerate their success in the clinic. Getting short on time, I think, so I'm going to skip a couple. I'm going to go pretty fast here and just highlight a second center that we also were able to gain funding for. Is focused on imaging. The Center for Multiparametric Imaging of Tumor Immune Microenvironments. It responded to a U54 call, and is led by Paolo Provenzano. It's currently in its early stages now. And we have 2 test beds, one led by Paolo, which is pancreatic focused. The other one is glioblastoma focused, which is led by myself and Dave Larga-Spada, and as well as the individuals you see listed here, who are key contributors in bringing this integrated engineering approach using imaging as a key tool. And it's in collaboration with the University of Wisconsin, and Kevin Eliseri being a key technology leader in this area as well. So here's kind of the outline of the C-MIghty Center, and allows us to gain data like this, where we can see green T cells navigating complex collagen-rich environments that you can see visualized, in which carcinoma cells reside, which are in red. And the T cells can go fast at times, but oftentimes just whiff and miss the cancer cells, or get stuck and not able to move in the stromal environment, negating their potentially beneficial effect. So here's kind of an outline of the core technologies there. And lastly, I'll just mention a new therapy modeling and design center that myself, Jasmine Fu, and Kevin Leader in the College of Science and Engineering are trying to develop, where again we can intake emerging therapies, bring mathematical modeling around them to try to optimize and design them in a way that's going to maximize their likelihood of success. We're currently trying to hire a scientific director and postdocs, and want to take a team approach to this, and we're currently developing a strategic plan, and we're grateful for funding again from the Cancer Center and the College of Science and Engineering to get this off the ground. So big picture, taking this initial grant, PSOC grant, and leveraging 2 more center grants, plus the launch of another center through internal funding so far, and launching a cancer bioengineering initiative where we take this engineering methodology of iterative technology development and design, and applying engineering technologies like imaging, modeling, genome engineering, microfabrication to accelerate that process and make them more likely to succeed. Thank you.
Thank you very much, Dr. Odie. So we've got time for one question. So whoever would like to be that person, raise your hand, please.
Great talk, Dave. Going back to the CD44 results that you presented midway through the talk, the biphasic behavior of CD44 and its implication in migration, would you predict too much inhibition of CD44 would be a bad thing?
Too much would be a bad thing? No, I think if you could totally kill it, that would be great. The problem would be is if you had patients who are sitting to the right of the optimum, like hyperadhesive, and treated them but didn't treat them completely effectively, they might end up kind of in between, and that is the worst case scenario. So you could actually harm patients if they weren't dosed properly there. So it's a risky strategy to take those patients and try to treat them with such a therapy because it has to be really effective. It'd be better to maybe just stratify them out, or yeah, it might be a safer move just to not try to treat those patients and focus on the patients who are in the middle, who are most likely to benefit. And if they're already low to begin with, then treating them isn't going to benefit them too much anyways, at least not this treatment.
Thank you.
Well, yeah, thanks for the invitation to share some of our work out of an ovarian spore in collaboration with the Mayo Clinic. So the title is Treatment of Advanced Ovarian Cancer Using CAR and K Cells. So really the impetus for this project was a call from the Mayo Clinic, who had held a ovarian cancer spore for about the last 10 years, and they were looking for new projects. And Tim Starr and Melissa Geller and I have been working together on some Randy Shaver funding for NKs for ovarian cancer, and so we put our hat in the ring, and after 2 submissions, the spore got funded. And so this was our original team, but actually, as many of you know, Tim is a lot younger than he looks, and so he actually recently retired, and so luckily Bo Weber was willing to step in and take his place on the project as another co-investigator. Additional collaborators here at the U of M are Jeff Miller and Martine Felices, who are, of course, world-renowned NK cell biologists, and also Dave McKenna, the director and head of the Molecular Cellular Therapeutics facility at our GMP facility in St. Paul, and then Scott Kaufman and Jamie Gomez, who are the main PIs of the entire spore. And so today I'm going to tell you about the thrust of our project. And so our overall goal is to develop, translate, and carry out a phase 1 clinical trial using a highly engineered allogeneic CAR and K cell therapy for the treatment of advanced ovarian cancer. And here in this diagram you'll see a couple times, but we're engineering in multiple different genes and receptors to enhance their function, as we'll talk about today. So first, why CAR NKs for ovarian cancer? Well, of course, inherently, NK cells have the ability to kill cancer cells using a number of activating receptors. They also identify MHC-low cells, which is typical of cancer cells, and can eradicate them directly. They also carry out antibody-dependent cellular cytotoxicity on cells that are bound by therapeutic antibodies or B-cell-derived antibodies. More recently, people have demonstrated that you can also equip NK cells with a CAR molecule to identify cancer-specific antigens, which will further enhance NK cell activation and mediate cell killing. Furthermore, in the last few years, engineering approaches have been developed for peripheral blood NK cells from many groups, including our own, and so that's readily available. Furthermore, work from a number of groups have shown that you can use ex vivo irradiated feeders to activate and expand peripheral blood NK cells more than 10,000-fold in the course of just a few weeks of manufacturing. And this, in the end, can produce an off-the-shelf therapy, an allogeneic CAR NK cell therapy that can be deployed readily for many different cancers. Moreover, they do not induce graft-versus-host disease, cytokine release syndrome, or neurotoxicities, which are exceedingly low with NK cell therapies compared to T cell therapies that are in the clinic. So effectively, they're potentially much safer than T cell therapies. And then specific to ovarian cancer is that ovarian cancer is susceptible to killing by NK cells, and the literature says that the percentage function and IL-15 responsiveness of ascites-derived or IP-derived NK cells actually correlates with improved progression-free survival and overall survival of these patients. And so as we looked at, the different sources of NKs that could be utilized for these therapeutics, people are using peripheral blood, iPS-derived, ES-derived, and even immortalized cancer cell lines. It's currently unknown how similar these are, and so much investigation is being put here, but we've really focused on peripheral blood NK cells as they are readily available from healthy donors. They are a true NK phenotype, if you will. They're non-transformed. We avoid the complexity of deriving them from iPS. They can be expanded in ex vivo, and they've been used in a number of clinical trials safely. So moreover, now we need to identify a cancer antigen to target with our CAR, and so we've chosen mesothelin as we think it's a good CAR target for ovarian cancer. It's basically expressed in more than 85% of ovarian cancers. It has low expression in tissues, shown here is just normal ovaries. And then it's also been used clinically, both as an antibody drug conjugate and as a CAR T product, both with relatively low toxicity. Another key aspect to using NK cell therapies is that historically their in vivo persistence has been very low. But recent reports using cytokine armoring, like the work shown here from Katie Rezvani's group, where they developed CAR-19 NK cells with built-in soluble IL-15, historically, NK cells in people do not last more than 3 weeks or so, but with this built-in cytokine armoring, she could detect at least the genomic DNA of her CAR NK cells for over a year, and more recently, this has also been recapitulated in other animal models. One other component that we've decided to include in our engineered NK cells is a synthetic receptor called RQR8. It acts as both a suicide gene and selection marker. And so the first component is an epitope from CD20 that allows it to be a suicide gene, such that if you need to kill the therapy in the patient, you can treat them with rituximab, which will induce cell death or targeting and elimination of this product. And also they have an epitope for CD34, which is really useful for GMP manufacturing because we can use this GMP Clinimax immunoisolation to actually enrich for the engineered cells. And so this is kind of the cartoon diagram of what we've been developing then. So it has the CAR, the soluble IL-15, and the synthetic receptor. We've developed a construct that expresses these from one single molecular construct that expresses the CAR, the RQR8, and the soluble IL-15. These are all driven by the strong synthetic MND promoter to actually express these in the NK cells. And so we thought about a number of different modalities for engineering, and we really landed on the use of DNA transposons, as they can also be utilized to engineer immune cells. And so if you're unfamiliar, these are a simple non-viral approach to engineering cells where you deliver your cargo as a DNA transposon plasmid and an enzyme called a transposase via electroporation. Once these are in the cell, the enzyme identifies your cargo of interest and integrates it stably into the genome in a random fashion, very similar to lentiviral retrovirus. This is not a new concept for engineering immune cells. There's been numerous clinical trials using the Sleeping Beauty transposon system, the piggyback transposon system, and the one that we've utilized called TC Buster was actually utilized recently at one of the local spin-ups here in town called Luminary Therapeutics, where they actually took something from concept to clinic, a CAR T product, in 18 months, largely because of the speed and efficiency of using this transposon system. And so other attributes of why we like these systems is that they mediate stable gene transfer like viruses. They've been used clinically. There's no limitation on the sequences you can use. There's this cargo capacity aspect of this where viruses have minimized cargo. Transposons, we can deliver large therapeutic cargos over 12 kb. There's some immunogenicity associated with viruses, but that's usually not the case for ex vivo modification. And then they're both randomly integrating, at least for lenti and retro. And really what we found in the literature is that transposases have a better integration profile than viruses. But the big point is the cost to manufacture is about 2 to 5 times less. And on the bottom, the efficiency now with some of these systems is actually very similar to viruses. Moreover, for our work, working on a small budget of a spore, is that we can get the GMP transposon in less than 4 months for $100,000 to $250,000, and the GMP transposase enzyme is readily available off the shelf for use. And so really what we've done over the last 2 years is generated a GMP-compatible manufacturing of CAR NK cells using this process. So in the first 12 days, we implement the engineering, which we typically can get anywhere from 50 to 80% of the NK cells engineered. We then put them into an enrichment to isolate our CAR-positive cells, and then do another expansion, which overall we can achieve more than 2,000-fold expansion in 19 days, and well over 80% of the NK cells express the CAR. So there are a lot of mesothelin CARs floating around in the literature, and so we had to decide which one we wanted to utilize for our trial. And so we identified two clinically proven binders, this SS1, FCFV, M912, and then a novel camelid-based binder. And so we wanted to really figure out which one of these CARs is maybe optimal for our application. And so again, we generated those DNA transposons that had the CAR RQR8 IL-15. And here's just a representative flow plot showing that, very high fraction of the cells are actually engineered, well above 85%. And then we took those and did co-culture activation assays with ovarian cancer cell lines. This is A1847, which is mesothelin high. You can see the SS1 and the 121 CAR produce more cytokine or better activated compared to the M912 CAR and also the mock NK cells alone. We also tested a mesothelin low cell line and saw similar results where the SS1 and the 121 CAR were inducing the most activation of these NK cells. We then took this into an in vivo model using IP injection of A1847 ovarian cells, waited 14 days, so ensured that there was ample engraftment of this tumor line, and then we gave one single dose of 5 million NK cells, either unengineered or engineered with the 3 CAR constructs. And then we did weekly imaging of these, and we also took blood at week 6 for analysis of NK cells in the peripheral blood, and then followed them till endpoint. And so here's the results of this. So on the left there, you can see the in vivo bioluminescent imaging, and it's pretty clear right away that the SS1 CAR is probably the winner. So these animals lived out over 100 days as compared to about 60 or 70 days with the other CAR constructs. And interestingly, when we looked at the peripheral blood, of course, we know from the literature that studies using NK cells in mice do not persist without support by a gamma chain cytokine. And again, here we have built-in soluble IL-15. And so you can see with our 121 CAR and SS1, we see robust amounts of NK cells in the peripheral blood of these animals, where we didn't observe this with the NK cells alone as expected. But kind of surprisingly, we also didn't see this persistence of NKs with the M912. I'm not showing the data, but also at sacrifice, when we study the IP fluid in the peripheral blood, these groups also still have abundant CAR NK cells in the animals. And when we kind of zoom in on the SS1 CAR here, you can see that actually one animal on the left was completely cured of their ascites tumor, and then 2 more of them appeared to have tumor. But we actually found that this is an artifact of the model, and that when we use a single needle to inject the tumor cells when we initiate the studies, you actually get some of the tumor cells caught in this IP lining where they cannot be kind of attracting NK cells, or they can't migrate to this region for some reason. And so now we've actually started using new needles for every injection, and we've resolved this artifact. And so we're running additional studies that should basically demonstrate complete cures, as we saw in the mouse that didn't have this artifact. So based on these in vitro and in vivo studies and other data I don't have time to show, we've decided to move forward with our clinical trial using this SS1 CAR. Some more specifics about kind of building up to this clinical trial. So we've partnered with the Be the Match International Bone Marrow Donor Program to identify our allogeneic source of cells. We plan to screen numerous donors that are recallable, likely more than 10 of them. We'll test engineering efficiencies, phenotyping, and functionally test them both in vitro and in vivo to identify optimal donors. And then clinical trial design and manufacturing. So the primary objective of this study is to assess the safety and determine the maximum tolerated dose of these cytokine-armored SS1 CAR NK cells. It is notable that they will be delivered via an IP catheter. They will also get preconditioning with Cy/Flu, and because we're using this catheter, we'll actually be able to take IP washings at various time points and look at the persistence of the NK cells and even their activation status from those isolations. As far as the manufacturing, we're assuming a maximum of 19 patients to be treated in this study. If we assume there's an average weight of 80 kilograms per patient, we can obtain CAR NK cells in enough quantity from a single apheresis donor to actually treat all 19 of these patients in our dose escalation study. And we're currently planning our technology transfer of the manufacturing process that I covered to our GMP facility in St. Paul by the year end. We also submitted a pre-IND packet in April of this year. Most of the questions were surrounding safety items with the manufacturing, looking at the transposon copy number, and also the source of the GMP plasmid and mRNA. There were also questions surrounding farm tox and if we could deliver these IP. Basically, all of the feedback was very positive and nothing that we didn't expect, and so we feel good about moving forward with these IND-enabling studies that are underway. So some conclusions and future directions. So DNA transposons are a safe, affordable, and rapid method to engineer immune cells. We've developed a GMP-compliant manufacturing process to generate our CAR NK cells. Mesothelin CAR NK cells are functional in vitro against mesothelin high and low cell lines. The use of a soluble IL-15 factor in our CAR NK cells allows for long-term persistence in vivo. These cytokine-armored CAR NK cells can cure ovarian cancer in some mice and significantly extend life in many. And then looking forward, so the next steps of our SPORE to get this clinical trial off the ground, we're doing comprehensive phenotyping and in vitro functional analysis of these cytokine-armored CAR NK cells. We're also performing additional in vivo studies using other xenograft models and even a PDX model through the ovarian cancer spores PDX core down at Mayo Clinic. And then, of course, we're transferring our GMP manufacturing process to MCT, where they will do process development, engineering runs, scaled runs, and QC testing for purity, sterility, and stability. That'll be done in 2024. And then we will finally file our IND for the phase 1 trial in 2024, and we hope to initiate and kick off the trial end of 2024, more likely 2025. And with that, I just want to thank everyone that's been doing this work in the lab and all of our collaborators here and at Mayo Clinic. Big shout out to the Randy Shaver Fund. If it wasn't for them, none of this would have ever happened, so thanks so much for your support, and of course, all of the support of the Cancer Center. And with that, I'll take any questions.
Thank you very much, Dr. Moriarty. All right, so we got time for one question. Who's the lucky person? Can I see a hand? There we go. Here, we'll switch.
That was really cool, Brandon. Quick question. How do you get around exhaustion when you do such huge ex vivo amplification before you put the cells back in?
Yeah, that's a great question. We haven't seen a lot of exhaustion. We need to do more comprehensive flow panels on, especially on cases where we're isolating from the animals. We have been able to see after tumor clear in other versions of this for cancer models in our lab that after the cancer is gone, the NK cells will persist in the animals long term. We've taken it out to over 200 days, and they can even be re-challenged with tumor and cleared again. And so the process we've developed, maybe it's the feeder cell activation or something, you know, the process is the product, as they say, and in this case, it seems to be working quite well. All right, thanks.
Awesome. Well, thanks for your question. So we're going to be turning it over to Carol Lang, Dr. Carol Lang.
Thank you, and thank you to everyone for staying here throughout the afternoon. We still have a great program left. I'd like to introduce our next session and our three speakers. This is the abstract awards session. We had 150 abstracts, and they were spread across our 6 programs. Program directors were asked to rank their top 5, and out of those 30, they were reevaluated and ranked, scored and ranked, to get our top 10% from those. And so I'm really pleased to announce we have Our first talk would be Daniel Chang from Zohar Sachs Laboratory. That'll be followed by Jock Lyman from Scott Dames Laboratory, and we'll end with Shui Wang from Ann Blaze and Anna Prisment. Each speaker will have 10 minutes to tell you their story, and we hopefully will have a few minutes for questions after each talk. Thank you very much, and so Daniel, take it away.
All right, Jackie's going to be the first one speaking. Thank you.
All right. Hi, everyone. Apologies for that little switch around, but my name is Jackie Lyman. I am a third year in the dual degree MCD-B&G and Genetic Counseling graduate program, and I'm excited to share some of my research today about a phase 2 clinical trial in prostate cancer. So prostate cancer is a very serious public health burden. It is the most commonly diagnosed male cancer, as well as the second leading cause of male cancer-related death. Prostate cancer is most often diagnosed at the localized disease stage, at which point most therapies are curative. However, a subset of patients will experience tumor recurrence, at which point the tumor is considered dependent on the androgen receptor signaling pathway. Therefore, standard of care therapy is androgen deprivation therapy. And though this therapy is very effective, a subset of patients will experience recurrence again, at which point tumor progression under androgen deprivation therapy conditions is termed castration-resistant prostate cancer. This has led to the advent of new potent next-generation anti-androgen therapies that have prolonged survival. However, recurrence is inevitable, and CRPC remains an incurable disease. So there is an unmet need right now for additional therapies for men with castration-resistant prostate cancer who are progressing on available therapies. So that brings us to a new therapeutic compound called sacituzumab govitecan, which is a novel antibody drug conjugate for the treatment of CRPC. So before I talk about SG, I need to talk about TROP2, which is the drug target of this ADC. So TROP2 is a cell surface glycoprotein, and the expression of TROP2 becomes more increased with advanced cancers like prostate cancer. In prostate cancer, the expression of TROP2 has been linked to aggressive phenotypes such as stemness, anchorage-independent growth, and metastasis. Additionally, the expression of TROP2 has been linked to the expression of androgen receptor variants, or ARVs, which are known negative prognostic indicators and drivers of treatment resistance. So collectively, TROP2 is an attractive drug target for aggressive and treatment-resistant prostate cancer. So sacituzumab govitecan is an antibody drug conjugate targeted against TROP2. So the majority of this compound is a TROP2 antibody conjugated to the payload SN38, which is a topoisomerase I inhibitor and the active metabolite of irinotecan. So upon targeting to the TROP2 molecule on the cell surface, the antibody drug conjugate gets internalized, releasing the SN38 molecule into the cell, where it can induce genomic damage and inhibit replication and transcription. So this brings us to the phase 2 clinical trial of sacituzumab govitecan in men with castration-resistant prostate cancer. So this phase 2 clinical trial is a multi-site collaboration. The PI of this trial is Josh Lang at the University of Wisconsin-Madison. We also have trial sites at Memorial Sloan Kettering and Weill Cornell, as well as biomarker studies being done here at the University of Minnesota. So the structure of this phase 2 clinical trial, enrollment criteria is being a man with CRPC who's currently progressing on standard of care anti-androgen like abiraterone or enzalutamide. Prior to treatment with SG, patients obtained a screening biopsy, and then SG was administered on day 1 and day 8 of a 21-day cycle. Following cycle 3, day 15, an on-treatment biopsy was taken from these patients, and then patients were followed for long-term follow-up. For the biomarker analysis, we recruited an initial cohort of 20 patients. From those patients, we obtained both matched screening and on-treatment biopsies from 13 individuals. Those 13 matched biopsies then underwent DNA and RNA sequencing, and I'll be sharing some preliminary results of this biomarker study with you right now. So first, looking at some outcomes from this clinical trial, one of the primary measurements of outcome was radiographic progression-free survival, which is essentially the time on treatment a patient experienced until their tumor progressed. So using a retrospective time point of 6 months, we stratified our 13 patients into our responder and our non-responder groups. So we had 7 of our 13 patients experience at least 6 months of radiographic progression-free survival. So the first question I wanted to ask was, how does the expression of TROP2, the drug target of SG, correlate to the clinical outcomes? So what we found was that TROP2's expression alone did not predict clinical outcomes on this trial, as we see by the comparable expression of TROP2 in both our non-responder and responder groups. And interestingly, the expression of TROP2 is maintained from the pre to on-treatment biopsies, indicating that, at least at the RNA level, TROP2 remains a viable drug target for patients being treated with SG. Another biomarker that I investigated was the presence of altered AR in these patients. So we know that alterations in the AR is a negative prognostic indicator and driver of treatment resistance. So looking at our DNA sequencing data, we identified AR gene structural variants in 1 individual in the screening cohort and 3 individuals in the on-treatment cohort. So not only were all individuals with an AR gene structural variant belonging to the non-responder group, but we also identified an increase in AR gene structural variants, indicating that altered AR could be a potential mechanism of resistance to SG. So taking this observation and testing it in our in vitro models, we used a cell line of CRPC called LNCAP95 that expresses both full-length and variant forms of AR. Compared that to the parental model that only expresses full-length AR, or culture conditions that isolate the signaling of the variant program, we're showing you dose-response experiments to the warhead SN38. And the takeaway is that the cell line that expresses both full-length and variant forms of AR is comparatively more resistant to SN38 than either model expressing one signaling pathway alone. So this is in agreement with what we see in some of the patient biopsies, suggesting that altered AR may be a resistance mechanism. So we repeated this experiment in another cell line that has been genetically engineered to express either full-length AR or AR variant alone, and we see a similar trend in which co-expression of full-length and variant forms of AR may be conferring resistance to SN38. So another biomarker that I wanted to assess was the DNA repair pathway, given the mechanism of action of the topoisomerase I inhibition. So looking at our gene expression data from a subset of DNA repair genes, I summarized here on the right and noticed a trend in which prior to treatment with SG, non-responding patients had a higher expression of DNA repair genes compared to the responding patients. And this prompted me to look more specifically at candidates within the DNA repair pathway, and I came across PARP1 as a potential biomarker of response to SG. So looking at PARP1's expression alone, I was able to stratify my clinical response in patients prior to treatment with SG. And what's interesting is that when you look at the screening and on-treatment biopsies, nearly all patients experienced this marked upregulation of PARP1 expression, indicating that exposure to SG may be inducing PARP expression, which may be grounds for combining SG with PARP inhibition to prolong therapeutic response. So this is an ongoing project. We have additional DNA and RNA-seq biopsies being processed, which will give us more power to address biomarkers in our data set. Ongoing efforts will assess synergism of SN38 with other partner compounds towards the goal of enhancing therapeutic application. We'll also be assessing TROP2 expression in our CRPC models towards the goal of moving these therapeutic regimens into an in vivo setting. So in conclusion, I've shared with you that SG is a novel compound strategy for treating incurable CRPC. I've nominated altered AR as a potential negative prognostic indicator and driver of resistance to SN38 and SG. We've identified DNA repair and specifically PARP1 as a potential biomarker and an emerging therapeutic vulnerability that could potentially prolong response to patients on SG. So with that, I would like to acknowledge the DAME Lab and our collaborators, and I'd be happy to take any questions.
Thank you, Jackie. Questions for Jackie? The question, can we get back there?
So I'm just wondering about the mechanism that links these AR variants with resistance. Is there any indication that these AR variants are driving increased DNA repair gene transcription at all, or how do you see this resulting in resistance?
Yeah, that's a great question. My hypothesis right now is the AR and AR variants could be intersecting with the DNA repair pathway to drive different expression profiles and potentially more DNA repair proficiency. But that's something that I'll be investigating in the lab for sure.
Other questions for Jackie? Thank you so much. Our next speaker is Daniel Chang from the Sachs Laboratory.
Yeah, I'll get it.
Hi, my name is Daniel, and I'd like to share with you all the work that we have been done so far to determine the mechanism of progression of myelodysplastic syndrome to acute myeloid leukemia. Myelodysplastic syndrome, or MDS, is a bone marrow cancer which has variable clinical presentation as well as patient outcomes. It is a malignancy characterized by bone marrow failure, which leads to anemia, bleeding, and they're more prone to infections. Unfortunately, one of the worst clinical outcomes for these patients is the transformation to acute myeloid leukemia, or AML. However, not all MDS patients will develop AML, so the risk of this leukemia transformation are associated with the genetic mutations that drive the MDS. So MDS is a mutationally heterogeneous malignancy where certain mutations can confer favorable prognosis, whilst some mutations like TP53 confer worst prognosis and the highest risk of progression to AML. And so you can see with the plot on the right, we see that the MDS patients with TP53 mutations, they transform to AML more frequently than the TP53 wild-type patients. However, like the molecular mechanism that drives the progression of TP53 mutated MDS to AML remains elusive. So to elucidate the molecular mechanism of leukemic transformation, we generated a mouse model of MDS that replicates the genetics that we commonly see in TP53 mutated MDS. We crossed a TP53 mutant mouse that models the TP53 mutation, and we also crossed it with a mouse model that models the deletion of the human chromosome 5q, which happens about 84% of the TP53 mutated MDS. And to model the genetic changes that transform the MDS to AML, we utilize Sleeping Beauty transposon system to mutagenize the mouse genome. Sleeping Beauty transposition is a well-validated method to introduce gain or loss of function alterations in mice. The Sleeping Beauty transposon harbors a strong promoter, which when inserted in a forward direction that an oncogene can be overexpressed. Conversely, when the transposon is inserted in the reverse orientation, the transcriptive gene can be interrupted and silenced this way. Specifically, genes that are disrupted that provides an advantage are expanded under this model, and the genes that are repeatedly inserted are what we would consider putative driver mutations. So after Sleeping Beauty transposition, our mouse model developed different types of leukemia that affects different lineages, such as T-cell acute leukemias, mixed phenotype acute leukemias, and of course, acute myeloid leukemia, which interestingly that we found are strongly associated with a TP53, strongly associated with mice that harbors the TP53 mutation. So in order to identify the genes that are frequently inserted or form fusion fragments with Sleeping Beauty, we perform RNA sequencing. We identify 61 genes that are commonly inserted across 29 mouse samples. And over here, each column represents a sample, and each row represents a fusion fragment that we were able to detect. And red means that the gene is fused. And we identify, something like ERG, ERAS, and IGROS, and which are genes that were found to be implicated in AML biology. So next, we looked at, how these fusion fragments or these genes that are being repeatedly inserted, how do they relate to TP53 status. And first, what we found is ERG are often fused with Sleeping Beauty in the TP53 mutated sample. And specifically, this fusion causes an increase in expression in the leukemias that were developed from this Sleeping Beauty ERG fused sample, suggesting that ERG upregulation may cooperate with mutant P53 to transform our mouse model of MDS to AML. So next, what we asked is, okay, well, now that ERG is Sleeping Beauty ERG fused samples have increased ERG expression. So how does that affect gene expression profile? So we performed pathway analysis or gene set enrichment analysis between the leukemias that developed from Sleeping Beauty ERG samples and leukemias that did not develop from Sleeping Beauty ERG fusions. And we found out that stem cell gene signatures as well as hypoxia signatures are strongly upregulated, and P53 mutations have been implicated to play a role in leukemia stem cell biology, as well as hypoxia gene signature are associated with poor prognosis in AML. So next, we asked whether or not high ERG expression causes similar changes in gene expression in human data sets. So we looked into TCGA AML data set as well as BAML data sets. And so what I've done is that I separated the tumors based off of their ERG expression, so just separating into high versus ERG low samples, as well as for samples whose karyotyping status is known, we will separate samples with amplified ERG versus those without. And then we perform, again, pathway analysis to compare human data with mouse data. And what we found is that our mouse data was able to recapitulate these gene expression changes between the ERG high samples versus the ERG low samples, or the ERG amplified samples and those that are not. Again, we've seen upregulation of the stem cell gene signatures, and then we also seen enrichment of hypoxia gene signatures as well, suggesting that our Sleeping Beauty mouse model and its the gene expression changes that it causes recapitulates what we find in human AMLs as well. So since increased ERG expression is associated with this stem cell-like gene signature, so we next looked into single-cell RNA sequencing data and to assess ERG expression in leukemia stem cells versus blasts. And we saw that the leukemia stem cells were expressed higher levels of ERG than the blast cells, suggesting that, again, like what we find with increased ERG expression is replicated in single-cell RNA sequencing data as well. And then lastly, we went back to look at the fusion data again, and what we found is that other than ERG, ERAS is also one of those genes that had common, that formed fusion common genes with Sleeping Beauty, and in fact, that SB-ERG and SB-ERAS, they happen to co-occur frequently together in the TP53 mutated AML, suggesting that those two might play a role together in cooperating with TP53 to progress MDS to AML. So this is just a rough summary, kind of what I'm doing so far. And so number one, I think what we showed is that Sleeping Beauty can disrupt enough genes to drive our mouse model of MDS to AML, and specifically that happens in the TP53 mutated mice. And then lastly, the Sleeping Beauty 4 genetic screen have implicated genes that have shown to play a role in AML biology, as well as genes who have roles to play in the progression of MDS to AML. I'd like to thank my mentors, Dr. Sachs, Dr. Largaspada, and Dr. Chad Myers, and the lab and the people who helped us collect the data. Thank you.
Questions for Daniel? Scanning. Dr. Dame has a question.
So ERG is a driver gene that's activated early in prostate cancer development, and so there are a lot of tools available for studying this transcription factor. Have you looked at any of this literature, and can you use any of those tools for next steps in your project?
Yeah, so ERG has also been implicated in just normal hematopoiesis, so it's not an unknown factor in terms of like how it's driving the progression. So previously, let me go back to this. So again, each column represents a sample and each row represents a gene, and red means that, okay, it's mutated or it's altered in the sense. Like our data is not clean enough where I can just say, oh, well, this one has the SB-ERG fusion, so let's look at the differential gene expression, because there are other genes that are mutated. So I would like to do that, to do more of like, whether or not the transcriptional targets of ERG are upregulated or downregulated, because ERG has both activation and like suppression functions. But I guess like the long answer or the short answer is I'll probably just generate a cleaner mouse model, maybe use lentiviral to overexpress ERG so that like the differential gene expression data would be cleaner in that way. Thank you.
Anyone else? Awesome. Thank you.
Thank you.
Our next speaker and final speaker is Dr. Shui Wang from the Blaze and Prismet collaboration. Thank you, Shui.
Hi, I'm Shu. I'm a postdoc at the Department of Lab Medicine and Pathology. My presentation topic today is about senescence-associated secretory phenotype, SAS-based aging index, and all-cause mortality in cancer survivors. First, a brief introduction to SASP. SASP is a phenotype associated with senescent cells, in which those cells release high levels of inflammatory cytokines, growth factors, proteases, and other factors to blood. And SASP is one of the mechanisms that contribute to aging and age-related phenotypes. So SASP proteins have the potential to be used as biomarkers for an individual's aging process. But no studies examined whether SASP protein-based aging index is associated with all-cause mortality among cancer survivors or not. So in this study, we aim to construct a SASP aging index and examine its association with all-cause mortality among cancer survivors. And in this study, we used the 72 SASP protein reported in Tunica 2018's paper to construct the SASP aging index. Our study population, we use the data from the atherosclerosis risk in communities study, ARIC. ARIC is a prospective cohort starting from 1987, and ARIC recruited white and black female male participants from 4 study centers, Minnesota, Maryland, Mississippi, and North Carolina. In ARIC, 5,000 plasma proteins were measured using the SomaScan assay from blood samples collected in 2011 to 2013, and this is the baseline for this analysis. Cancer cases in ARIC were ascertained using state cancer registry, and deaths were ascertained through the linkage to National Death Index. Using the ARIC data among participants with protein data available, we identified 3,699 cancer-free participants and 806 cancer survivors. In this analysis, we define cancer survivors as survivors who have survived for more than 2 years at their blood collection to minimize the effect of active cancer treatment. To construct the SASP aging index among cancer-free participants, we randomly select 2/3 of them and use them as the training set. The training set were used to construct the SASP aging index. And then the remaining 1/3 cancer-free participants were used as test set. The test set were used to validate the SASP aging index. Construction and validation of the SASP aging index. Using the training set, we applied ridge regression to train the 72 SASP protein against chronological age. And the SASP aging index was constructed as a weighted sum of proteins. And then we apply the SASP aging index to participants in the test set to validate the index. We validate the SASP aging index by calculating its correlation with chronological age. The Pearson correlation coefficient between the SASP aging index and the age was 0.52 in the test set. And then we apply the SASP aging index to cancer survivors and calculate the age acceleration for the SASP aging index. The age acceleration for the index is the deviation of the SASP aging index from age. And in this analysis, we estimate age acceleration as the residual after regressing the SASP aging index on age. So the age acceleration captures the effect of SASP aging index independent of age. Our analysis method in cancer survivors. We examine the distribution of demographic, lifestyle, medical factors across age acceleration in cancer survivors. And we also applied Cox proportional hazard regression to examine the association between age acceleration for the SASP aging index and all-cause mortality until 2019. In this study, we report a hazard ratio per 5-year increase in age acceleration. And the model was adjusted for demographic information, BMI, smoking, alcohol intake, hypertension, cardiovascular disease, diabetes, and kidney function. Some result. This table shows the distribution of baseline characteristic across the tertile of age acceleration for the SASP aging index in cancer survivors. And we found cancer survivors with the highest age acceleration, they tend to be current smoker. And those survivors were also likely to have a lower physical activity level and to have cardiovascular disease and lower eGFR, which capture their kidney function. And the survivors with the highest age acceleration, the mean of their eGFR level is below 60. This slide shows the result for the association between the age acceleration for the SASP aging index and the all-cause mortality among cancer survivors. In this analysis, we found age acceleration was statistically significantly associated with all-cause mortality in all cancer survivors. The hazard ratio per 5-year increase in age acceleration was 1.6. And we also found that age acceleration was significantly associated with all-cause mortality in breast cancer survivors and colorectal cancer survivors. The association in prostate cancer survivors was positive but did not reach statistical significance. We also examined whether the association with all-cause mortality in all cancer survivors was modified by sex and race. And in this study, we did not detect any interaction with sex and race. The p-fold interaction for sex and race was greater than 0.2. Summary and conclusion. In this analysis, we found cancer survivors with higher age acceleration for the SASP aging index were more likely to be current smoker and to have cardiovascular disease, lower physical activity, and lower eGFR level. And in the study, we also found the age acceleration for the SASP aging index was associated with an increased risk of all-cause mortality among all cancer survivors, breast cancer survivors, and colorectal cancer survivors. The association in prostate cancer survivors was positive but did not reach statistical significance. In summary, our results suggest SAS protein-based aging indexes show promise in mortality prediction in all cancer survivors, breast cancer survivors, and colorectal cancer survivors. Our next step is to validate the SASP aging index in other data sets stratified by cancer treatment and stage at cancer diagnosis. Acknowledgement. I would like to say thank you to both of my mentors, Dr. Prisman and Dr. Blaze, and I really appreciate their help in my work and life. And also, I would also thank Dr. Guan. He always answers my statistical questions of my project. And also thank you to our two wonderful research assistants, Simon and Zexi. They have put a lot of effort into this project. And thank you to ARC participants and staff for their contribution to the ARC study and the ARC Cancer Group led by Dr. Plus. This study was funded by National Cancer Institute. Happy to take any questions.
Questions for Dr. Wang? I have one. So are there ways to modify the aging index for healthy people, such as exercise? Have you done those kind of studies to say, can you lower yours if you have a high one?
Yeah, so we create this index. We hope this can be used to measure people's aging process, and then some people with the same chronological age, they may have different biological aging process, and there's some actually anti-aging interventions. So if people draw their blood and then we measure they have a higher age acceleration, maybe they can have some anti-aging intervention do some activity or take some medication to decrease their accelerated aging process.
Excellent. There's hope then for all of us. So Dr. Yi has a question.
So if I understand your cohort correctly, there are a group of patients who eventually had a cancer diagnosis but did not survive for 2 years. Have you ever looked at them? I'm just wondering if the aging index is a feature of cancer itself or a feature of the cancer treatment those patients have received who survived.
So this index is actually construct in cancer-free people. So we assume like we can measure like some cancer-related like aging induced by cancer, and we exclude those survivors who did not survive for 2 years because we are kind of afraid that they are under active treatment, and active treatment will influence their protein level. So that's why we actually we exclude them. But our next step is actually kind of Erica is actually working on the cancer treatment data, so we do want to like consider treatment data. This is kind of our next step. One of them.
Thank you so much. So let's give all three speakers a great big round of applause. We have poster session number 2.
Well, first, after a very hard process of counting the votes, we've got the winner for the Faculty Flash Talk competition, which is, can I get a drum roll, please? That's pretty good, I like that. The winner is the Transplantation and Cellular Therapy Program, Dr. Sorko. Nick, where are you at? Somewhere over there? Yeah. That's awesome. Well, there you have it. So yes, now we're gonna head off into our second poster session of the day now. So presenters, if you'd like, you can go get your posters, which have been hung up if you drop them off at the registration table. And then at this poster session, we're having a whopping 4 of our top 10 abstracts. So there's a lot to look at for this session. So enjoy yourself, and we will resume back at 3:35. Thank you. All right, everybody get back to your seats. We are going to kick off our final session for the day, which is Unraveling Genetic and Cellular Mechanisms to Tackle Cancer with Dr. Scott Dem and Dr. Anna Prisment. So up first is going to be Dr. Dem, and he will present as a member of the Division of Molecular Pathology and Dynamics. Thank you.
All right, I was told the slides were ready to go. Okay, there we go, perfect. Thank you very much for sticking around after the poster session, and I'll try to keep us on time so we can get out of here and or see some more posters and then get out of here. So thank you very much for the invitation to come tell you about our collaborative work. Graduate student in my lab, Jackie, already gave a very nice introduction to our work on prostate cancer. And so I just want to emphasize that localized prostate cancer can be treated with surgery or radiation. My lab studies the metastatic form of prostate cancer, which requires systemic therapy. With prostate cancer, that systemic therapy is usually hormonal therapy, specifically to inhibit the effects of the androgens testosterone and dihydrotestosterone. And so that can be achieved by inhibiting testicular androgen production or synthesis, and also through the use of potent androgen receptor antagonists like enzalutamide, apalutamide, or darolutamide. And the main challenge facing our field is that prostate cancer initially presents as a castration-sensitive disease, so these therapies are very effective. But with time and under the suction pressure of these therapies, prostate cancer will progress to an advanced and lethal castration-resistant stage. And so we study the mechanisms of this progression. And so one needs to look no further than the target of these therapies, the androgen receptor, to uncover a lot of really interesting resistance mechanisms. So these hormonal therapies exert a lot of selective pressure on the androgen receptor protein, mainly via alterations in the gene encoding the androgen receptor, the AR gene located on the X chromosome. So there are mutations that have been described in the AR gene. This affects the cell's responses to anti-androgens and castration-based therapies. The AR gene in an upstream enhancer can be amplified, and this occurs frequently in advanced prostate cancers, leads to overexpression of the protein. And then my lab has uncovered AR gene rearrangements or structural variants as frequently occurring in the genomes of these advanced prostate cancer cells. And a major emphasis of our lab's research has been to understand the role and potential therapeutic significance of these AR gene rearrangements. In a subset of cases, we've been able to show that AR gene rearrangements can promote synthesis of altered variant forms of the androgen receptor protein. And so this is a very simple cartoon where there are some AR gene rearrangements that have affected the 3' end of the AR gene. We've identified these in tumors from patients as well as models such as patient-derived xenografts. And we've been able to use genome engineering approaches to create these AR gene rearrangements de novo in cell-based models that would normally express full-length AR. So for instance, here is a prostate cancer cell line called R181 that expresses the full-length androgen receptor. This is an androgen-sensitive prostate cancer cell line. And when we engineer a deletion or an inversion into that cell line as observed in patients, we can convert that cell line to one that expresses an AR variant. And the important thing about these AR variants is they retain the parts of the androgen receptor protein that are important for its activity, but they've lost the ligand binding domain. So they've become insensitive to androgens and insensitive to anti-androgens. And so thereby they can drive resistance to therapy. We've been profiling AR gene rearrangements in many different sources of tissue. And so in castration-resistant prostate cancers, we see that AR gene rearrangements occur in anywhere from 20 or 30 to 50% of advanced prostate cancers. It really depends on the therapy treatment status of those tumors. So these are frequent in castration-resistant prostate cancer. And what I'm going to tell you about today are 3 vignettes describing 3 collaborations that we have designed to study the clinical and therapeutic significance of these AR gene rearrangements in castration-resistant prostate cancer. So the first collaboration that I'll tell you about is a collaboration with investigators at Duke University, Andy Armstrong and Susan Hallaby. The second collaboration that I'll tell you about is a homegrown collaboration with Stephanie Huang. And the third collaboration is another homegrown collaboration with Emmanuel Antonarakis. So for this first collaboration, we've obtained plasma specimens from patients that were enrolled in a phase 3 clinical trial. And this phase 3 clinical trial was the Alliance A031201 trial. So this was a trial that randomized over 1,300 patients to receive enzalutamide or enzalutamide plus abiraterone. See, these are all AR-targeted therapies. And the hypothesis was that the combination of abiraterone and enzalutamide would provide a greater survival advantage than enzalutamide alone. This turned out to be a negative trial, but it also turned out to be a rich biomarker resource, which we've been able to leverage for our studies. And so using that plasmid DNA, we were able to obtain DNA sequencing data from the cell-free DNA of 776 of those patients. And the DNA sequencing that we performed involved a targeted panel that investigated the androgen receptor gene, as well as several additional genes known to be frequently altered in advanced prostate cancers, so we could understand the genetics of resistance to therapy, but then potentially also the genetics of response to therapy. And so what we did is we took these 776 samples and rank ordered them left to right by decreasing circulating tumor DNA fraction. So in the cell-free DNA from the plasma of these patients, we can detect genetic alterations that are coming from the prostate cancer cells in those patients, and that's a measure of circulating tumor DNA, and we can quantify that. And so you can see that a subset of the patients in this trial have very high circulating tumor DNA, and in this schematic here, we drew a cutoff at 20%. So everything above 20% we're considering frank circulating tumor DNA positive, and in those samples below 20%, unknown. And so what we can see is that there are alterations in AR, specifically gain of the AR gene body, gain of the AR upstream enhancer that are detectable in these cell-free DNA samples. The majority of the alterations are occurring in those samples that are frank positive for circulating tumor DNA. But you can also see that a subset of these samples seem to have positive ctDNA signal, even though those samples are predicted to have very low or undetectable circulating tumor DNA. We are able to detect AR gene rearrangements in these samples as well, and they're occurring with equal frequency in those tumors or in those samples that have high circulating tumor DNA, as well as those samples that have low circulating tumor DNA. And then we can also profile AR mutations. So we can get very deep information about alterations in the AR just from a simple blood sample from these patients. And this is significant because when we perform Kaplan-Meier survival analysis of the patients on this trial, we can see that those patients that have frank positivity for circulating tumor DNA have the worst outcomes. But this green line represents those patients that had below 20% circulating tumor DNA, which is at our threshold of detection, but were positive for alterations in AR as well as other genes such as MYC and MYC-N. So even in those samples that are low or perhaps negative for circulating tumor DNA, if we can identify these genetic alterations, that's a prognostic biomarker for poor outcomes. And in those patients that have low circulating tumor DNA and undetectable alterations in these genes, those are the patients that did the best in this trial. So we continue to profile samples from these patients at progression on the trial, and also are asking about correlations with these specific genetic features and outcomes. The homegrown collaboration with Stephanie Huang is intended to address the problem that those tumors that have AR gene rearrangements or AR structural variants have the highest AR activity, despite therapies being designed and being used to inhibit that AR activity. This was a very serendipitous collaboration. It just so happened that Dr. Huang had attended a seminar from a graduate student in my lab, Mark Daniel, as he was giving a seminar for the Microbiology, Immunology, and Cancer Biology graduate program. She approached us afterwards and thought that some of her work could be applied to our problem in prostate cancer. And so we did just that. We applied for an internal grant from the Office of Academic and Clinical Affairs, which was awarded back in 2019. And the goal of this grant was to take Dr. Huang's expertise in computational drug prediction profiling. So specifically what she does is she can take advantage of DNA or RNA sequencing data and drug sensitivity data from big cell line profiling studies and use that to build models that can predict the sensitivity of cells to therapy based upon that RNA-seq data. With our patient data, when we have RNA sequencing, we can use that to predict the drug sensitivity of individual patient tumors to individual drugs, and then we can stratify patients and tumors based upon features that we're interested in to potentially predict sensitivity. So since 2019, we've been collaborating extensively, holding biweekly joint lab meetings, collaborating on various studies, and these are some of the key outcomes to date. We've published a couple of collaborative papers, and we've obtained NIH grant funding in the form of 2 R01s to support our collaborative work. And I just want to highlight a recent publication in PNAS where Dr. Huang was able to identify COL3 as a potential drug that would have increased sensitivity in prostate cancer patients that have the worst overall survival time. And so she was able to rank tumors based on their RNA-seq profiles from high to low sensitivity to various different types of drugs, and COL3 turned out to be a top hit in this trial. And so what we were able to do is use our prostate cancer cell line models that represent sensitivity or resistance to AR-targeted therapies to test whether these drugs have different effects. And so COL1, for instance, seemed to have the lowest IC50 or the highest potency in our cell line model that represents resistance to standard of care therapy, as opposed to the parental cell line, which is sensitive to standard of care therapy. And similarly, in an apoptosis assay, the prostate cancer cell line that represents resistance to standard of care therapy was the most sensitive to this drug. So what is the mechanism by which that is occurring? Well, it seems like COL3 is able to inhibit the expression at both the RNA level and at the protein level of the AR variant that is expressed in the cell line that's resistant to standard of care therapy. Whereas in the parental cell line, the full-length AR mRNA and the full-length AR protein is insensitive to this drug. So this is really exciting as a basic scientist, where a computational biologist can give you a list of drug candidates and drug leads that may have differential sensitivities based on the biology of the system that you're studying. And in this case, COL3 turned out to be a very interesting hit. And so we're moving forward with this into preclinical models and then hopefully eventually clinical trials. A final homegrown collaboration is with Emmanuel Antonarakis, who's a genitourinary medical oncologist here at the University of Minnesota. And it is based upon our observation that those tumors in castration-resistant prostate cancer patients that have high burden of AR structural rearrangements are those that have very high copy number of AR. So these amplification events in AR and rearrangement events in AR seem to go hand in hand. And we recently published a paper demonstrating that AR amplification on extrachromosomal DNA or extrachromosomal circular DNA is a likely mechanism for this occurring. So in a subset of advanced prostate cancers, the AR gene can actually leave the genome and be captured on extrachromosomal DNA circles. And we've been able to show that this is a mechanism by which the copy number of AR can increase to exceptionally high levels. The AR copies that are there are structurally unstable, so they can accumulate a high number of AR rearrangements. And this allows the tumor cells to make not just more AR, but more types of AR, including these AR variants. And so we've hypothesized that this situation where prostate cancer cells have dramatically upregulated the levels of AR could actually represent a vulnerability for a paradoxical treatment approach that Dr. Antonarakis has pioneered both here at University of Minnesota and previously at Johns Hopkins University. So all prostate cancer therapy is really designed to turn the AR off by suppressing androgens or the use of anti-androgens that inhibit AR. A paradoxical approach is the use of high-dose androgens to treat advanced prostate cancers. Perhaps the resistance mechanisms that castration-resistant prostate cancers accumulate may make them vulnerable to this high-dose therapy. And so this is achieved by injection of testosterone at biweekly or tri-weekly intervals. And what this leads to is a high spike in androgens levels within the serum of the patient, but then it declines back to castrate levels. And this is achieved sequentially via these testosterone injections. And in a subset of patients, this therapy is actually clinically effective. And a major question in the field has been, what are the features of the tumors that actually determine sensitivity to this paradoxical high-dose bipolar androgen therapy? We've hypothesized that AR on amplification on extrachromosomal circular DNA may actually be one of these vulnerabilities in tumors to bipolar androgen therapy. And we've been studying a tumor model, a patient-derived xenograft called LuCap105CR, which has accumulated very high copy number of AR on extrachromosomal DNA. And when we take these tumors and grow them in mice and then begin treating them as we would clinically with high-dose testosterone, you can see that these tumors respond very favorably to that therapy, but will eventually accumulate resistance, as all advanced stage cancers eventually will. But you can see in these tumors that have relapsed in response to AR-targeted therapy, or in response to this bipolar androgen therapy, we see a downregulation in the burden of these AR extrachromosomal DNA molecules. And so by toggling between high-dose testosterone therapy as well as anti-androgens, we may be able to control the copy number of these extrachromosomal DNA molecules and thereby extend the duration of response to therapy in these patients. And so this is something that we're moving forward with both in a clinical trial as well as our preclinical models. And with that, I'd be happy to take any questions about any of the work that I presented today. Thank you.
Right, nice talk. I was wondering, related to the second part, the computational approaches, could that be used to sort of determine whether patients with castration-resistant prostate cancer have one or multiple pathways of resistance, just to determine whether they may need a combination therapeutic approach or just one?
Yeah, so I presented just one way in which Stephanie Huang's lab is able to predict sensitivity to drugs. Another project that her lab has been working on, that we've been collaborating on as well, is predicting combinations of drugs that may be more effective in patient tumors, model systems. Basically, any samples that you have, whether they're clinical samples, patient-derived xenograft cell lines, for which there is RNA sequencing data, Dr. Huang's lab has algorithms that can predict drug sensitivities based on any biology that you would be interested in. It's really quite remarkable technology that she's been able to develop.
Just quickly, based on that data, do you see in patients multiple pathways of resistance, or is it mostly just one or two?
Oh, there's multiple pathways of resistance in the patients that we've been studying. So AR alterations are the most frequent in castration-resistant prostate cancers, but P53, RB1 alterations, as well as many others, have been implicated in resistance to therapy.
That looks like that's what we got. So I'm going to welcome Dr. Prisment up to the stage. Thank you very much.
Good afternoon. Thank you for staying here for such a long time, but it's the last presentation for today. And so I will be talking about proteomic aging clock before and after cancer diagnosis, and my postdoc has already started talking about one of the aging index that we created. I will talk first a little bit about our study team, as I was requested to do so, and then briefly give a background on chronological age, biological age, and cancer, the interrelation, and discuss how we developed proteomic aging clocks, and then how we tested association between proteomic aging clocks and cancer risk and outcomes in cancer survivors, and briefly discuss future directions. Here is our multidisciplinary study team, which is very collaborative, very enthusiastic. I'm very lucky to have these people on my team. I'm a molecular cancer epidemiologist myself, but although I have a minor in biostats, it is my first experience doing machine learning, and I'm very grateful that having Dr. Guan, biostatistician, on our team, and he's also had expertise in creating aging clocks, and very good that he had already a lot of experience working with epidemiologists, so he knows what we need. That was very important. Certainly we had an oncologist on our team and researcher in cancer survivorship and outcomes, Dr. Ann Blaze, and another member of our team, Bharat Yagarajan, who has expertise in measuring and analyzing age-related blood biomarkers. And when I invited these people to be on the grant, on the proposal, yes, everybody was excited. So I hoped that that was a sign that the idea was worth it. It was very interesting. And also our team in the University of Minnesota is strengthened by researchers from other universities, from Johns Hopkins, Elizabeth Platz, renowned cancer epidemiologist, and from UCSF, Dr. Peter Gans, who has experience in studying proteomics. These people helped us not only with their scientific expertise, but also provided access to the large data sets that we needed. And finally, but I cannot help but mentioning our excellent graduate students in epidemiology, Shu Wang, who just presented today, and now she is postdoctoral researcher, and a graduate student in biostatistics, Zoshi Rao. And so only due to this team, I think it was possible to do our study. And so this is an important topic now, because you know that our society is aging and getting older, and age is the greatest risk factor for cancer. And on the left, you can see how most of the chronic diseases, the death from them increases with age. And the second line from the top is for cancer. And so, not only the population is aging, but in line with this, the number of cancer survivors is increasing gradually. And so it is expected to be 26 million cancer survivors in 2040. Imagine just this figure. But the good thing that they live longer because of better treatment and of diagnosis is at early age due to screening. But these cancer survivors experience a lot of comorbidities, perhaps at younger age than cancer-free persons. So the role of age is very important in cancer survivors and even before cancer. So what is the relation between aging and cancer? So as many of you heard, so some aging occurs due to cancer therapy. So cancer survivors say that they feel older than the people without cancer of the same age, and also not only they feel older, but some biomarkers show that they're older. But cancer itself can promote aging due to features of cancer such as inflammation, oxidative stress, and different lifestyle factors, for instance, obesity and lack of physical activity. Both aging and cancer have similar hallmarks of aging, so that's part of it, that cancer affects aging, but also aging even before cancer can contribute to the development of cancer. So it is important to measure these associations, but it is difficult to measure it because age itself, you know, is not modifiable. It's just the time since birth. So the concept biological age has been introduced. What I see here. Yeah. And so it estimates the state of health, that an accumulation of all comorbidities, that during time, that of the damage, the biological damage that happened. And so it can be older than chronological age or younger. And this deviation between biological age and chronological age is called age acceleration. But again, the question is still there, how we will measure biological age. So one of the things that was proposed is to using physical performance or just frailty as a biomarker of biological age. Another approach is to use biological markers measured in blood. And these are called aging clocks, and they have been created based on epigenetic, transcriptomic, or other omic biomarkers. And perhaps you heard about epigenetic clock that can assess your biological age. It's weighted average of age-related CPGs, and there are clocks by Horvath, Hannum, and more modern clock by Belsky. And you can actually go to the website, pay $500 and measure your biological age if you're interested. I never tried it. I'm scared. But epigenetic clock, although they're most studied, they have certain limitation. Underlying mechanism is not known. There is large technical variation between samples. And for some reason, there is very poor correlation between different types of clocks. So we proposed another approach, and it was first proposed not by us. It's just to measure proteomic aging. There were several studies recently that to create aging clock based on proteomic biomarkers. And proteomics is a little bit behind epigenetics and transcriptomics due to the technology being behind in this area. But recently, several proteomic-based assays have been created, such as Olink and SomaScan. So we created proteomic aging clock as a sum of protein, is weighted sum of proteins, and the advantages compared to epigenetic clock is better reproducibility, and they reflect phenotype a little bit better, and the function of protein is more clear, of proteins is more clear than epigenetic markers. And also, different proteins may be targeted using anti-aging drugs. To our knowledge, no studies examined the association between proteomic aging and cancer risk or outcomes after cancer diagnosis. I would not talk much about atherosclerosis risk and community study because Shu Wang already introduced the study. I would also like to mention that you can see that it's called atherosclerosis risk, but it actually was transformed into cancer study. So it's a very interesting approach. And so it has all the biomarkers already measured for cancer, for cardiovascular disease and other chronic diseases that can be applied in the studies of cancer. And overall, there are more than 5,000 cancers that occurred till 2015 and more than 2,000 cancer deaths, if somebody is interested in this study. So they measured 5,000 proteins in a little drop, 55 microliters in frozen samples 3 times. And the strength of this assay is it can measure proteins a very different range of concentration in the same sample and very low detection levels. But the limitation is the proteins are measured in relative units, so they need to be validated. So here is the hypothesis and study aims for our funded R01, and the hypothesis that higher proteomic aging clock acceleration is associated with cancer risk and worse outcomes in cancer survivors. And I apologize, sometimes I call this acceleration PAC acceleration, sometimes PA acceleration, so it's the same thing. I will describe study aims about which I will be talking today, and we tested associations between proteomic aging clock and cancer risk in ERIC, and then we validated them in another cohort, multi-ethnic studies of atherosclerosis study. And in the second aim, we tested associations of proteomic clock with mortality and morbidity, such as frailty and dementia in cancer survivors. Proteomic aging clocks were constructed similarly to the methods that was described by Shu Wang. So we use machine learning methods and at middle age, applied to middle age and late age persons, and created published clocks and de novo clocks in ERIC. This is associations for clocks in ERIC at mid-age people and at those later age, and it shows the correlation between the proteomic aging clock on the y-axis and chronological age on the x-axis. And you can see that correlation is pretty good at both times, on the left in red in mid-age, and in the right in blue for late age. We also reproduced the study in MESA by applying clock in mid-age, ERIC mid-age clock, and for some reason, the correlation became even higher. It's hard to explain because it's different population, but we're working on this. It became 0.9. So we found that people with high acceleration were more likely to be women, had low education, never drank, and had worse kidney function. Yes, some results are very interesting about never drinking and age and later age. So we showed that age acceleration is associated with cancer incidence and mortality, and this is a complicated graph. So the first line shows cancer risk, and the second line cancer mortality, and we saw weak associations for both for cancer risk and for mortality, and one means that everything to the right means statistically significant, and it's hazard ratio, HR, which is similar to relative risks, but in longitudinal studies. We also saw associations for colorectal cancer, for both for risk and mortality, and for lung cancer. And the associations existed among never smokers for mortality. So it means that it's not that smoking that promotes this association. We looked at association between acceleration and cancer mortality by sex. and found that that female, for some reason, have higher, stronger association between PAC and cancer mortality for all types of cancers under study, and for colorectal cancer, it was the difference between men and women was statistically significant. So we concluded that PAC is a robust measure of biological age, and it's strongly correlated with chronological age. It was significantly associated with risk and mortality of all cancers combined and cancers of lung and colorectal. But it's not clear why it's for these cancers, but not for breast or prostate, although it is in line with previous study on epigenetic clock. We also look at the association of PAC acceleration with mortality and in cancer survivors, and also saw association among all cancer survivors, breast cancer, and colorectal cancer survivors. We did not study lung cancer survivors because of lack of power and because they died before, many of them died before diagnosis. There was association with dementia risk and stronger association with frailty, and all associations were multivariate adjusted and statistically significant. So we saw association with several types of cancer, with dementia and prevalent frailty in cancer survivors. Our next step would be to reproduce these results in MESA and also to compare these results for epigenetic aging clocks. So far, epigenetic clocks were measured only in a small number of participants in ERIC, but we see it shows that the association for proteomic clocks are a little bit stronger. But in the future, it will be measured in everybody. It will be pretty close soon. So our more long-term goal is to create the biological measure that may be easily assessed in clinic. These proteomic aging clocks are great, but they include like 500 proteins or even 1,000 proteins. So it's hard to imagine how they can be measured in clinic. More biologically based markers, the one that Shu Wang showed, based on senescence, can be more appropriate, or I will show you very quickly another potential aging metrics based on 9 biomarkers associated with mortality that can be very easily measured in clinic during simple clinical visit. And we started exploring these aging characteristics and even submitted a paper in Health and Retirement Study. This is just prevalence of cancer, cross-sectional study. And here, significant associations are all over this line. So we saw association with colorectal cancer prevalence and lung cancer prevalence. It's interesting, these are the same cancers for which we saw associations with proteomic age. And so we hope that in the future we will create biological aging clock that will allow us to identify cancer patients that will be able to determine how much therapy would be needed. It could also serve as surrogate endpoint in clinical trials of anti-aging drugs, not to wait for the outcome. And if there is a causal relations with cancer or other disease, which is most likely because these are the same pathways, by targeting biological aging, it would be possible to target the disease. But these are just dreams. Yeah, I would like to thank you, our team that I mentioned at the beginning, and researchers in the ERIC, in the MESA studies, and the participants in both of the studies. Thank you. Now questions.
Any questions? Any questions? We got one?
Was there another? Okay. That was really, really interesting. Thank you. We're doing some similar work in companion dogs, but looking at it from a slightly different perspective, where we're looking at what we think is the aging environment and how that influences risk of the development of cancer. So we're trying to see if picking up an aging phenotype will predict risk of cancer. And I think one of the things that we wrestle with is, you mentioned you can go to the website and measure your aging clock, but you haven't done it because you're afraid. And so we have that same concern, right? That we can provide an answer of cancer risk or presence, and if we don't have an intervention, then all we do is create anxiety. So do you have any sense for, and we certainly have some ideas, but do you have any sense of effective interventions that can change the rate of acceleration or that can even reverse it? And I think Carol asked that same question earlier. So
and we just talked to Dr. Melissa Geller about potential intervention that she's doing. Yeah, I don't know. I don't see her here now. And so, yeah, but intervention. Oh, sorry. Yes, yes, there are some potential interventions that now there are multiple clinical trials. Yeah, if you look at this clinicaltrials.gov, it's most of them are looking at metformin and rapamycin. Yeah, and I also know there is a study going on in our university, and I forgot his name, Dr. Puskaric, I think. Do you know him? He's studying senolytics in relation to septic shock, actually. So, but he's just studying, but he has funding to do this. So we are also planning to do, but have not decided what.
maybe we should talk some more.
Yes, that would be great.
Any other questions? There we go.
Hi, thank you for that talk. I'm wondering, when you see this kind of association, how do you decide which one is the cause, which one is the consequence? So this could be, you know, some kind of predisposal to cancer that lead to those accelerated aging theme. Like, what do you chase down the road when you have these highly correlated traits and both are very complex?
So I'm just thinking how to better explain. Certainly we are not able to ascertain, to determine causality. But the sequence is pretty clear because we are measuring aging clock at in 90s. In people who are free from cancer, for instance, if we are talking about cancer risk. And then we are following them for 20 years. So there is no way that cancer could cause this age acceleration. And so we're also accounting for all potential risk factors. Well, not all, but for many lifestyle factors, including age, actually. So all our associations are independent of chronological age. And finally, we have a testing set, or a training set, where we create this clock in cancer-free people. So, and then we apply it to the testing set, and then validate. Yeah, I actually didn't present the results on cancer risk in another study on external validation, and we can see that the associations are still there. Yeah, so certainly it's not causality, but it's the best we can do without clinical trial.
Awesome. Well, thank you for your questions and your awesome presentation. Appreciate that.
Thank you. I will show you one picture since we talked about our team. And this is the last presentation. And tomorrow is Halloween. So, this is part of my home team. Oh, sorry. Here, there. Yeah.
It's very cute. All right, I'd like to invite Dr. Carol Lang back to the stage to wrap this all up.
Thank you. Well, we're nearing the end of the day. We have one more poster session and a reception. And this doesn't need to be a half an hour, but I've been asked to kind of summarize some themes from today and then maybe open a discussion for future directions. And I've asked Dr. Lagerspada to help me do that. So I've put him on the spot. In listening to the talks today, I had some themes I jotted down. Starting with the 10,000 families, I think that Jen showed us how to look at innovation and innovative ways to engage our communities. We learned about how you can do that from a simple idea that became a very large cohort that I think will provide a legacy and nucleate many, many, many projects. I think that's only the tip of the iceberg. That starts with gathering data for the types of exposures that are relevant to Minnesota and our communities and how they impact cancer risk, and then raising the awareness for the types of exposures that are relevant to our catchment. And we saw how even having lunch, sometimes it starts with just an informal lunch, so there needs to be some food, and just being open-minded to talking to your colleagues, and exchanging ideas. And Dorothy showed us how this lunch between her and a basic scientist could result in this huge seed that went into this huge tree that became even a forest and went all the way to the whole United States and into changing policy. And again, that is a huge legacy, I think just the tip of the iceberg for many, many more projects that have involved many other careers and engaged many other scientists. The complexity is there. Sometimes we start with this food and then we think, okay, now we got to have some money. So usually that's a pilot grant from the Cancer Center, an internal award, an external award from Randy Shaver, an RFA from the NIH, which provides the money. And then usually there's even more food and bringing people in, recruiting your colleagues. I think that this nucleation stage requires a vision to see things a little differently, and that's why we cross fields, because we each have a different sort of place that we sit and how we see things. It takes a lot of patience, institutional patience, persistence. We saw how Dave Odie submitted that grant 3 times before they got it, and there was even dancing involved after that, and more food probably. So this vision, this patience, this persistence, this dedication, and this environment, really, that we have. And Dr. Paskett's talk, I really think that what came out of it for me, where I sit, is I was like, well, how do we get from one person, one patient, to a whole population? And then how do we bring that back to the basic scientists and say, like, what is the epigenome? What is the epigenetics of social isolation? What is that doing to the transcriptome? How do we engage more basic scientists in population science to try to get at the actionable mechanisms? We have interventions to reverse things at the social sciences level, but we also need to know what are the actionable targets that are happening when someone is stressed. So for me, I kind of looked at that. I think that one thing that came out of today too was this, we had kind of all levels of people involved across all programs. We tried to show a snapshot of teams that formed that were successful. These involved innovative technologies we saw with Dr. Moriarty's talk and Scott Dame's talk, these innovative technologies that have come in. Sometimes you just see a talk and you have an idea, and then it takes one more step to invite that person to lunch and reach out to that person. And I think the Cancer Center provides that environment for us in so many ways. And I hope that we can get back to that robust interaction stage that we had before COVID. I think it's been a challenge post-COVID to do that. So I encourage all of you to try to, not only when you have an idea, to articulate your idea, develop your idea, reach out to players, and see if you can make a seed, nucleate a seed, and then see if you can look for an RFA or some money to do that, and grow that up. First of all, it's very stimulating. It's something we can be really proud of as a cancer center, that we have all these amazing teams and such a supportive environment. And so I would like to thank all of the organizers of today, most notably Laura Klein, and all of our sponsors and exhibitors, everyone who presented, including all the poster presenters. And I'd like to invite David up here to talk about some of the maybe future directions we could talk about as a group and ideas we might have for where we need to go. One place I really think we need to go, we saw this aging in cancer, but let's also talk to our endocrinologists who work on diabetes and obesity and metabolism. Because I think one thing about these teams is they see big things. They try big things. They see a bigger picture, not so myopic. They see into other cancers and past their little project into a much bigger arena. And I think that we have the environment to do that here. We have an amazing Department of Medicine, an amazing division of endocrinology. We have a lot of focus on aging now. It's a buzzword, and we could bring in so much common science around those three things. So that's one thing I sort of got out of it, is this aging theme needs to transcend, more than cancer. So, David.
Yeah, absolutely. So I think it is interesting to note that aging is the biggest risk factor for cancer, and yet we, don't typically study it. We don't study it in the laboratory very much. We don't study it with our research animal models. And that brings up the bigger theme that we have an incredible group of epidemiologists in our cancer center, and I think that hypotheses from epidemiology need to be tested for biological plausibility in the laboratory, and that kind of work is highly fundable. And our keynote speaker gave an incredible example of how stress could impact cancer development in a rodent model, and yet there aren't very many examples of work like that. I think it's something that would be highly fundable by scientists in this cancer center if we took our work and we cross boundaries and disciplines. I think we could do that quite a bit more. And it's equally important for new technologies. So for a lab like mine that does genomics to try to do proteomics, like we just heard about from the last speaker. So those are some themes that come to mind in thinking about what came up today. So yes, for those of you who work in a basic research lab, I think it's really important that you try to find new opportunities for research that are inspired by questions that the epidemiologists bring up, or that you get by talking to clinicians, which you can do at meetings like this, about what patients are really experiencing, their patients, what's really happening to them, what kind of factors we're not studying in the laboratory that we probably should be. So do we want to open it up for comments?
Yeah, I'd love
questions?
to hear from anyone in the audience who can add to that or has other ideas and what you got out of today. I'm staring at some people.
Yes.
Lots of people raising their hands
That's
right now. Holy crap.
I think they all know that
We're all
there's
tired.
open bar.
The bar will be
That
open
is coming
soon.
quickly. We got anybody. This is your opportunity to ask a question
How
to
about
marvelous people.
Dr. Anton Arakis, just sitting there with her.
I got you.
There you go. If I know your name, you're in trouble.
Well, thanks to both of you for that great summary. I was in clinic today, and I missed the first half of the session, but what I did catch was very inspiring. And as one of the few clinicians maybe in the audience, and I often have PhDs or graduate students or undergraduate students coming to me and asking if they can work in my clinic, and I always oblige them. And one of the things that I always ask them to do when they go back to their labs is to take one thing that they've learned in the clinic, or one patient that they've seen, or one problem that they've seen, even if their mentor is not specifically working on it, going back and trying to think outside the box and working on that. So I would encourage any of the undergrads or postdocs who have had clinical experience or contacts to reach back out to their PIs and say, hey, can we work on something a little bit different? It might be outside of the PI's comfort zone, but it may address a real clinical question.
Thank you.
Should we let him go?
I guess we should let you go.
All right, well, I just want to say one last thing at the end because, and that's really to, again, acknowledge everything that Doug Yee has done for this cancer center. He announced earlier that he was stepping down as cancer center director, and he didn't give us a chance really to congratulate him for 16 years of amazing leadership, where the cancer center just got better and better, and 4 CCSG renewals. So give him a hand. Thank you, Doug. Yes, stand up.
Speech. All right, with that, we will transition into drinking.
Yeah, three sentences, and then we're going to be heading off. So a big thank you to Teleflex for making this reception possible. So a quick round of applause for them. There it is. All right, so poster presenters, it's your opportunity right now to get up, get back to your space, get ready. Everybody else, you can follow suit. As you heard, there's some drink tickets, should be in here. And then you also have some appetizers as well. So make sure to go enjoy that, mingle, talk to each other, discuss, and enjoy the rest of the evening. Thank you very much.